Some places, the bottom is covered with what's called pteropod ooze, like inches. Not so much here, it seems like. If you ever have a chance to see them, just know they're phenomenal animals. They're transparent, which is a trick midwater animals mastered. And they're just effortless swimmers. Of course, they kill and eat other animals, but everybody does down here. Because what else is there? Unless you're living in the dirt, eating whatever falls next to your borehole, like some of these guys are. You can see the holes and all the scrapings. Those are animals who sediment and scrape the newly deposited stuff toward them to eat. Life's hard. But if you're under an area that's had magnificent upwelling and all sorts of productivity, you might win big. And if it's seasonal, you can always count on next year. It's kind of the long, slow road to life. There's more pteropod shells, heteropod shells. Huh. That's kind of interesting. You know, on first glimpse, it looked to me like a vertebra, but I don't think so. Everybody wants to find like the fossilized whale vertebra. This isn't going to be it. Don't think. Maybe the sediment just boiled up and then froze. No response from the geophysicists. Oh, do you think the sediment's so hot it just boiled up and those things froze when they hit the cold seawater?
I don't know. I think it's cold here, actually.
Really?
It looks cold.
All right, well.
But if it's a recharge spot,
it should be cold. That looks like a big disturbed area. Our friendly brittle star, Xenophilophores, stalked crinoid. Another stalked crinoid. Looks like a burrowing anemone there, just left at center. The pretty. I guess that yellow is the yellow sponge we've been seeing. I found a little bit of something hard to latch onto. I don't recall we've seen anything else yellow.
What do you get?
Our friend Mr. Shrimp. Looks a lot more serene than those shrimps that were hassling the octopus eggs yesterday. No, dive before. I get nightmares looking at those shrimp. They're just mean.
It wasn't my favorite thing to look at.
But we think of shrimps as nice, you know? Rats never hurt anybody. Then you see them in action. Your view changes. Can't judge a shrimp by its cover. All right, well, what we'll be doing later in this dive is more heat flow probes. As long as we're in sedimented areas where we can collect the data, we need to. Once we hit the rocky area, we'll be exploring, hunting, looking for octopods so we can have a party, and, you know, hoping for the best, exciting stuff. An anemone down there, it's pretty cool, and I think this is an isopod. Whoops, you may not be seeing this, just dancing. Just amazing little beasts. I assume that's feeding behavior. Each of those tentacles seems to have extensive filaments reaching down, and they could move like that to sweep anything in the water column toward their mouths. Looks like a not really efficient way to forage, but hey, it's a living, so it must be good. Woo-hoo. That one looks a little more menacing. That one could have its own dark theme music. That crest on its head, those eyes. Hide your children. Uh-oh. Somebody asked at one point why animals have such bright colors. You have to realize that water does funny things to the spectrum as we see it. Red, the color, is produced by wavelengths of red being reflected from something. In the water, if you're, 10 meters away from something red and look at it, it looks black. So you might as well be red as black, because it'll have the same net effect on anything close to you, close enough to each of that is. Whew. The amazing thing is when you're down here with an ROV focus at different depths, you see things that, you know, if you've been on trawling cruises, if you've been through Alvin, you just never even imagined were out there. How can you have a 10-foot long transparent thing like that hover? It was a friendly rat tail. Heat flow probe 1 will be transiting about 100 meters to waypoint 2 once everything's back on board the ROV SuBastian. And we look for more excitement.
How far do you want to go for the next one?
Waypoint 2.
Waypoint 2? Okay.
Yeah. We'll do standard imagery transects.
Okay.
Now, you know, let me know when you begin.
Hey, on the bridge, put an updated waypoint for you.
Updated waypoint. What sort of speed you want me?
Point 2, please.
Understood.
All right, during the video transects, we'll be going ahead at a steady speed at set altitude, not collecting or even zooming, but we might indicate where hotspots, or pardon the term, maybe to come back to.
Really shadow. Move the arm out. We are moving at point 2 at 1 meter off.
Transect begun.
Set the shadow. And now that came up. So I just move it out to the starboard. Tighten the arm. There we go. That's better.
Yeah, have you done it since you started actually on the live one?
What's that?
The shadow.
That way it's still behind us. Yes. Stop my picture.
That's probably better them follow us than the other way. You know what's odd here? I think we've seen 2 brittle stars. Yesterday, there were scads of them. Okay, 3. This area's got a lot more info on it too. A lot more animals boring into the sediment, burrowing into the sediment. More of those worm tubes. crab with an anemone. So, going.
Hey, folks. I can hear a little bit of a weird stutter on our audio. Oh, there you go. See, it's dropping out. I'm gonna reset our whole system here, so it might buffer for a little bit, but we will be back. can still hear a bit of a hitch in our audio, so we're going to keep trying to troubleshoot that on our end.
Good morning, everyone. My name is Beatriz Naranjo from Universidad de Costa Rica. It's a pleasure to join you this morning. We are now exploring small Caballito, which means small horse. And our objective here is primarily to do heat flow measurements and also some transects on the abyssal plains. Right now, we are about 3,000 meters down the surface. We are in an area 200 kilometers off the coast of Costa Rica from a place called Guanacaste in the northern Pacific of Costa Rica. I'm joined here by Dr. Miguel Semedo, City of Porto, Portugal. He's a microbiologist. He's conducting some experiments, and he will be helping me to take some notes of the dive. And also, I'm joined here by the PhD student Rob Perrin from the University of who is in charge of the heat flow measurements. Buenos días, mi nombre es Beatriz Naranjo de la Universidad de Costa Rica. Es un placer estar hoy aquí con ustedes narrando por la siguiente hora. Estoy acompañado el doctor Miguel Semedo, que es un microbiólogo de la Universidad de Porto en Portugal. Él está realizando investigaciones en microbiología y va a estar ayudándome tomando notas del buceo. Y también estoy acompañada por el estudiante de doctorado Rob Perrin de la Universidad de Canadá, que se está encargando de hacer las mediciones de flujo de calor, que es uno de los principales objetivos que tenemos en el buceo de hoy. Entonces vamos a estar explorando un lugar conocido como Caballito. Es un lugar que está bastante cerca del ensayero. Y además de las mediciones de flujo de calor de las que tenemos objetivo, vamos a estar haciendo transectos, videotransectos, para poder determinar la densidad de organismos en esta área. Como ven, ahorita estamos haciendo la medición, así que esto tarda varios minutos. Vamos a estar deteniéndonos cada 100 metros y realizando una prueba de estas en los primeros 8 sitios. Esta es la segunda. Así que vamos a ir avanzando un poco lento durante la mañana, y en cuanto terminemos vamos a seguir explorando, buscando señales de agua caliente. Cualquier pregunta o comentario en inglés o en español pueden hacerlo a través del chat, y el grupo aquí de científicos y científicas a bordo intentará responder sus preguntas. También me acompaña, por supuesto, los pilotos del ROV, que son parte importante de esta expedición. So we will be doing heat flow measurements in each waypoint. which is separated between each other by 100 meters. And this is the second one that we are making. We will do 6 more, and then we will start searching for venting and exploring all the area. So right now we have a cool spider right there. We've seen a lot. Think so. Oops, other way. I'm just learning how to control the camera, sorry. Really nice animals. I remember when I was at the university studying, my professors say that we didn't have this in our country, or maybe that they would be really small. And now we know that that was not true. So it's pretty amazing to be able to see all these animals living here. Not my hour, I promise. I I don't know if it says spider, right? I don't know. Yeah. Okay. So now I'm joined also by Dr. Rachel Lower. She's from Calgary as well in Canada, and she's been doing lots of model right before this expedition, and it was really amazing because her models show that we might find inside where we were yesterday. So it's pretty amazing to see all these computing models in real life, and she was right, or the models were right. I love Ahora me acompaña también en la sala de control la doctora Rachel Luer de la Universidad de Calgary en Canadá. Ella ha estado realizando muchísimos modelos por varios años en su computadora, y la razón por la que ella hace estos modelos es porque es geofísica y estaba buscando señales de dónde puede haber flujo de calor de acuerdo a los estudios que hacen con la corteza terrestre. Y es bastante emocionante ver cómo todos estos modelos computacionales se traducen en datos reales que podemos probar en campo. Como comentaba en inglés, es la primera vez que controlo la cámara, así que espero mejorar un poco al final de la hora de mi turno. Para aquellos que nos han estado siguiendo, han notado que cada hora tenemos una científica diferente. Eso se nos ha permitido también narrar en español. Es algo que nos ha emocionado mucho. Poder comunicarnos con costarricenses y latinoamericanos, latinoamericanas y personas de habla hispana de otros lugares del mundo. Estos organismos, si no me equivoco, son del grupo de los octocorales. Hemos estado viéndolos en fondos suaves. Estos sitios se conocen como planicies abisales. Y algo interesante que nos van a permitir todas estas imágenes es comparar estos lugares con otros ambientes similares en el mundo, principalmente la zona de Clarion-Clipperton, que es una de las zonas más estudiadas y de las que se encuentran más cerca de nuestra región. Este es un sitio entre las zonas económicas exclusivas que pertenecen cerca de Hawái y México. Y tenemos la doctora Diva Amon, que ha estado trabajando muchísimo en esta zona, donde además hay varios contratos de exploración para realizar minería. Así que comparar todas estas planicies nos va a ayudar a entender mejor lo que tenemos. Mucho de lo que estamos viendo es nuevo. Bless you. Aquí tenemos otro pequeño coral. For the first time that we see images of our abyssal plains, and all this information and footage will help us to understand more about them and also compare them to other areas to our country. For example, the abyssal areas of the Clarion-Clipperton zone. Un saludo a Kimberly hasta Panamá. Muchos chicos alrededor del mundo nos han estado siguiendo. Muchos colegas y muchas colegas. Hoy estamos teniendo algunos problemas con el audio. El equipo técnico está intentando resolverlo, así que si se escucha un poco mal, pedimos disculpas. Esperamos que esto se pueda resolver en el transcurso de la mañana. Comentarios de otros colegas en Latinoamérica que han estado también teniendo la oportunidad de estudiar sus ambientes de aguas profundas. Esperamos que a nivel regional podamos comprenderlos mejor. Estudios en Colombia, hay bastantes estudios también en la zona cerca de Galápagos y como comentábamos las personas en las transmisiones, el Falcor 2 va a quedarse en la región por unos años. Así que estamos seguros que después de estas exploraciones vamos a tener una mejor comprensión de toda nuestra región en la zona profunda. Yeah, that one. A little bit to the left.
Oops.
There, yeah, thank you. Este organismo que estamos viendo en el centro de los láseres, para que tengan láseres de color verde, tienen 10 centímetros de distancia entre ellos. Y esto nos permite realizar estimaciones del tamaño de los organismos. Justo en el centro del láser hay un organismo que está con célula y que pueden llegar a ser bastante más grandes que el que estamos observando. Yeah. So we just finished the heat flow measurement here, and we will be starting our way to our waypoint number 3 with the lasers on.
Keep them.
Yep.
Further away, so like 70-ish.
Such.
No, further north. That'll work.
Right there.
Yep. It's really on there.
Acabamos de finalizar el flujo de calor en este sitio. Vamos a avanzar poco a poco hacia el siguiente punto de observación, número 3 de nuestra inmersión. Cada sitio está distanciado por 100 metros y entre cada uno de ellos vamos a realizar datos que nos van a permitir hacer conteos de todo lo que estamos observando. Así que vamos a avanzar lentamente a 1 metro de altitud del fondo. Para aquellos que se están uniendo en este momento, que estamos explorando una zona aguas en las aguas frente a la provincia de Guanacaste, a unos 200 kilómetros de distancia y aproximadamente a 3 kilómetros de profundidad. Hace unos 10 años aproximadamente se visitó esta precisamente por estas señales de flujo de calor que indican mucho sobre nuestra corteza terrestre y cómo ésta se está enfriando. Toda esta zona es una zona volcánica. Todos los montes submarinos y montículos más pequeños que estamos estudiando se generaron en la dorsal del Yeah. So, let me know when you start.
When I get back over to the actual line, that a little bit.
All right.
Going.
Okay.
Morning.
I just wanted to double check. Are we doing
We
1 meter of a bottom,
1 meter. We are just moving to the
0.1
waypoint.
meters per second speed, no zoom, tilted to just above the edge of the basket.
Yeah.
Okay, and lasers.
Yeah. We because
No laser.
no laser, so we are repeating that.
I mean, we can still use it for qualitative, but
Oh yeah.
yeah.
But we will repeat this. I mean, this was not planned, but we will do it. All right. No, this will be from waypoint 2 to waypoint 3.
So they moved to the but that's how they were going to repeat. Like,
Oh.
what's up?
We're coming back to waypoint.
Come back over to this.
No, waypoint 2.
Yeah, just getting back on the line as I was south of it.
Okay.
But now I'm gonna make that.
Precisely, yeah. Thank you. como les comentaba, esta zona que estamos explorando se generó en las, bueno, las partes rocosas se generaron en la dorsal del que es una zona donde divergen las placas tectónicas y permiten el ascenso de magma, y este poco a poco se va enfriando y ha ido migrando hacia donde está en su posición actual. Después esto se va cubriendo de sedimento, y parte de lo que estamos haciendo, interdisciplinario que tenemos, incluye muchas personas del área de biología principalmente, pero también de microbiología, de geología, de paleobiología, perdón, sí, paleobiología específicamente. Y también tenemos geofísicos y hidrogeólogos a bordo. Ahorita nos estamos devolviendo nuestro punto exacto, el número 2, para empezar el videotransecto en la línea que tenemos marcada como ruta para hacerlo. Vamos a estar haciendo alrededor de 6 mediciones más de flujo de calor, y entre cada punto donde hacemos las mediciones de calor hacemos un videotransecto. There we go. Okay. Start. Yeah.
With point 2 right now.
Alright. I think if you can go slower.
Okay, let's go to point 1 right now. Okay, so our speed is right there,
Alright.
and our altitude is right there.
Okay. So we are.
So you want to do 1.1?
Yeah, that's what Diva just told me.
Okay.
With the lasers on?
With-
Oh, look at that, they're on. I mean, flames are coffee.
Okay. I have one here. Yep.
Only one is really, really
Yeah.
slow.
Okay. Let's move it to 1.2.
Like this.
Yeah.
Alright.
Yeah.
Earlier in the cruise,
Yeah.
so it's-
I don't know. Diva have the wrong information. Este organismo que tenemos en pantalla es miembro del grupo de los octocorales, un grupo bastante interesante. Tenemos a bordo a la doctora Dalis Cabridi, que ha trabajado mucho con este grupo, principalmente en aguas someras, realizando buceos con tanque, pero poco a poco, gracias a la tecnología y las colaboraciones que hemos podido realizar a través de la Universidad de Costa Rica con otras instituciones, explorando cada vez más profundo y conociendo mejor toda esta fauna que estamos viendo. Este es un grupo bastante interesante no solo por su biología, suelen albergar otros organismos en ellos, sino también por sus propiedades químicas. Muchos estudios se han realizado y se siguen realizando con organismos de este tipo, como octocorales, también corales negros y esponjas, para permitir la búsqueda de medicinas, tratamientos contra el cáncer, por ejemplo, y muchas otras. Es parte de la importancia de todo este mar profundo. It doesn't say, but it's 1.1, so I think we're good here. Yeah. En pantalla tenemos un erizo de mar, uno de los grupos que también hemos estado observando muchísimo, así como estrellas y ofiuras. En este momento no vamos a cambiar la cámara, queremos que todo el transecto tenga el mismo ángulo y el mismo aumento para poder tener datos comparables. En algunos otros segmentos de la ruta que tenemos vamos a estar haciendo exploración entre los puntos que no tienen videotransecto, así que en esos casos sí vamos a poder estar haciendo, aumentando el campo para poder observar con más detalle los organismos. Hemos visto muchos cangrejos que tienen anémonas o zoántidos encima algunas veces. Estas son relaciones mutualistas que le permiten a las anémonas o a los zoántidos, que también son parte del mismo grupo de los cniarios, transportarse y tener mejores oportunidades para capturar alimento. Por ejemplo, el cangrejo se beneficia con una forma de camuflaje, y tomando en cuenta también que tienen como un escudo protector encima, ya que estos organismos suelen tener células urticantes. Han sido bastante los que hemos visto. Empezamos a ver pepinos por acá, que también ha sido uno de los grupos donde nos hemos enfocado, no solo en los videos, imágenes, sino también en la recolección de organismos para poder estudiarlos con más detalle. Tenemos a bordo a Holly Lutz, asociada al Field Museum de Chicago, que ha estado haciendo investigaciones con microbioma, toda la comunidad de microorganismos que viven dentro de otros organismos y que componen, por así decirlo, como de genes que le permiten a muchos organismos realizar funciones. Muchos organismos inclusive han perdido, por ejemplo, funciones en su intestino, ya que esas funciones las pueden cumplir microbios que viven dentro de ellos. Y es parte de lo que estamos haciendo. Cuando regresan las recolecciones, que los organismos recolectados, en el laboratorio se realizan disecciones de estos para hacer estos análisis y después analizarlos con metagenómica de vuelta en la costa. De lo que estamos viendo aquí son los xenofióforos que les estaba comentando. Es increíble que sean organismos de una sola célula. Son células multinucleadas, eso sí. Y en algunos de los que hemos recolectado hemos encontrado comunidades de otros organismos vivos. En una expedición pasada inclusive encontramos huevos de peces. Estos tubos que vemos aquí son tubos de poliquetos, de gusanos. Nuevamente un saludo a todas las personas de Costa Rica que nos han estado siguiendo, sus mensajes de motivación y agradecimiento. Para nosotros ha sido una experiencia increíble poder compartir con un grupo de ciencia, no solo de una gran calidad científica, sino de una gran calidad humana.
It's been 0.1, 0.2 right now. It's
This was like the magnetic. It's going to be a really good thing.
So they say that they've been doing this at 0.2.
months of like weeks ago we did it here. The speed, yeah.
Speed of a
See where we are now.
Yep.
Yep.
What's the question?
Because they were saying that it was 0.1. It's like
Oh, no.
really slow.
Yeah, it has been 0.2.
Okay.
Yeah.
All right.
We are going at 0.2. Right?
Yeah.
Well, it depends on what the if I have to move this to the left or right, but it's the speed right here. So.
Yeah. The important thing is to write down what you did.
Yeah, yeah.
Yeah, I think we originally had thought we would start with that.
Yeah.
But I think on the last track, also that was what we were at.
I always thought it was 1 meter up, 0.2 on our speed.
Yeah.
Yeah.
Okay.
Yeah.
All lights on, lasers on, just above the.
Yeah.
Okay.
Okay. Thank you.
What?
So we're almost getting to our waypoint 3, where we will be doing some more heat flow measurements. As we go with the transect, we are not able to zoom in any organism. So in this way, we can have a consistent way to compare our information from the images that we are obtaining.
We will be broadcasting our midwater.
Okay.
Unless something
Yeah.
unexpected happens.
All right. So yesterday, those who joined us were able to see the midwater on the way while SuBastian was going up ship, and today we hope to be able to do that again. Yesterday we had really nice, really cool encounters, so hopefully we will do that again. So stay with us. We will be doing some more heat measurements on the first part of the dive, but then we will be the outcrops and looking for shimmering water and exploring the fauna that we are going to find. Una parte muy importante del trabajo que hemos estado haciendo, aprovechando que tenemos un gran equipo de personas que hablan español en el barco, alrededor de la mitad hablamos español, es conectar con de hemos estado conectando con estudiantes de diferentes colegios y escuelas y ha sido una de las partes más importantes que hemos hecho. Hoy, por ejemplo, vamos a estar conectando con un colegio de la Cruz de un colegio de Tarrazú y un colegio de la zona de Osa en el sur del país. Personalmente son todos los lugares donde he vivido en algún momento de mi vida, así que estoy muy emocionada. En el caso de Tarrazú, pues conectar en el que tuve la oportunidad de estudiar. Y han sido experiencias muy bonitas poder enseñarles el barco. No solo el trabajo científico, sino también el trabajo de los artistas que tenemos a bordo, que nos han estado inspirando y inspirándolos a ellos, haciendo pinturas increíbles. En este momento ya terminamos el transecto, vamos a realizar otra medición de flujo de calor, así que vamos a alrededor de 15 minutos más. En estas mediciones de calor lo que se busca es entender si la temperatura del sedimento está por debajo o por encima de los valores esperados. Los valores esperados, aquí las personas de geofísica lo saben de acuerdo a la edad de la corteza terrestre. Y en base a eso, si la temperatura es menor, pueden tener un indicio de que el agua se está filtrando o recargando como si fuera un acuífero hacia la corteza. Y si la temperatura es mayor, más bien se está descargando agua. La razón por la que cuando se descarga es más caliente es porque esta agua atraviesa la corteza terrestre y en ese proceso se calienta debido a que se encuentra más cerca de la tierra y ese calor se comparte hacia el agua. Y estas señales que estamos buscando son pues bastante importantes porque son las señales que buscamos para encontrar dónde están esos flujos de agua saliendo que hemos visto muy asociados a las comunidades de pulpo que encontramos. Ayer fue muy evidente. Teníamos sitios llenos de pulpos justo donde el agua estaba saliendo, ya que esto también mantiene las rocas limpias, que es necesario para que los pulpos peguen sus huevos a las rocas. Metros de ese sitio donde no había agua emergiendo de la roca, no teníamos pulpos y teníamos las rocas con sedimento. Así que todo se combina y es personalmente y para todo el equipo creo una de los aspectos más interesantes de poder aprender de tantas personas a bordo con diferentes experiencias. Aquí tenemos una comunidad de zoántidos viviendo en lo que probablemente es un coral muerto, sobre lo que es un coral muerto, y una anémona en la parte superior. We are seeing a group of zoanthids living on a dead coral, and on top we have a really cool anemone. We'll stay here for about 15 minutes while we do the heat flow measurement. And right now we are at our waypoint number 3. We are exploring about 1.5 kilometers of this place called Caballito, which means small horse. Yes, exploring a site to the east of this place that we are exploring today. The xenophyrophores that we've been seeing a lot on these abyssal plains. On the back, we have an umbellulid octocoral. We've been seeing a lot of these shrimps with long legs, and we also are seeing a polychaete, a worm, in this tube. Right now, we are checking our heat flow measurement or equipment because we are having some inconsistencies on the data. Ahorita estamos cerca de finalizar nuestra expedición. Tenemos el buceo de hoy y otros 3 buceos más. Ha sido pues una experiencia bastante increíble. Esperamos tener descubrimientos importantes también en los próximos buceos. Entre los principales hallazgos o logros de la expedición hasta ahora se encuentran, por supuesto, haber confirmado que los huevos de los pulpos que encontramos y que se encontraron hace 10 años en el Dorado Outcrop son viables. Eso es pues un dato bastante interesante, ya que es el 4º lugar en el mundo donde este evento. Cuando empezamos la expedición era apenas el 2º. Sin embargo, en la misma semana que estuvimos explorando el Dorado, se encontró otro sitio en Canadá donde se encontraron pulpos también asociados a flujos de agua caliente. Y pues encontramos el sitio que exploramos ayer, que está hacia el oeste de donde estamos ahorita. Así que en una sola semana se descubrieron 2 sitios nuevos donde tenemos estos grupos de pulpo incubando huevos. Y ahora 4 en el mundo, y que 2 de esos se conocen de Costa Rica. Ese es uno de los principales hallazgos. Otro hallazgo importante ha sido el descubrimiento de un sitio importante para rayas de profundidad, para una especie que se conoce como Pacífico, una especie que hasta hace algunos años era bastante rara de observar, pero poco a poco, conforme vamos explorando, nos damos cuenta que son organismos bastante, pues no muy abundantes, pero que sí suelen visitar algunas de estas áreas. Encontramos un submarino conocido como Tengo Set, con muchísimas cápsulas o huevos de estas rayas. Tuvimos la oportunidad de analizar un par y confirmar que son también huevos viables. Esta es una de las rayas que logra vivir a mayor profundidad de todo el grupo y con periodos de incubación bastante largos, que estimamos entre 4 años, posiblemente más. Así que son especies, al igual que los tiburones con los que están emparentadas, bastante vulnerables por su lento crecimiento su tardía y por supuesto el hecho de que el huevo tarde tanto en desarrollarse es otro de los elementos que hace que sea tan importante en estos hallazgos. Se habían encontrado hace unos años en Galápagos, también asociados a flujos hidrotermales, otro sitio de importancia para su reproducción. Y precisamente también esta semana, en el mismo lugar donde se encontró el nuevo sitio de pulpos, en Canadá se encontró uno para rayas de otra especie. Pero bueno, ha sido una semana importante de exploración y descubrimientos en el mar profundo. I'm seeing here a lot of fans of the ascent. I am as well. It's one of my favorite parts of the dive. So we hope that we will be able today to broadcast also the returning of the SuBastian ROV. Ayer tuvimos interesante, vimos muchísimos organismos, principalmente plancton gelatinoso, como tenóforos, medusas, y esta parte del buceo no siempre se transmite. Ayer se hizo y estamos en aguas increíbles, hemos visto muy espectaculares y hoy esperamos poder transmitir en vivo también el ascenso de SuBastian al barco. En unas pocas horas vamos a estar también explorando cosa de este pequeño monte submarino, así que los invito a quedarse con nosotros. Vamos a estar teniendo cada hora una persona diferente liderando el buceo para combinar un poco las explicaciones en inglés y en español, y también que un poco más de las personas que tenemos a bordo, que como les comentaba tienen conocimientos distintos de diferentes elementos o áreas del mar profundo. También les invito a visitar la página de Schmidt Ocean, donde hemos estado subiendo blogs, donde se explica con más detalle, tanto en inglés y en español, todo el trabajo que se realiza a bordo. Y por supuesto las redes sociales, donde compartimos pequeños videos con los principales momentos o momentos más emocionantes de todo lo que hemos observado. Yeah, we collected some octopus last night on the way, and yeah, we were able to see them alive. Was pretty amazing. They were from the genus called Japetella, which are known to be associated with the minimum oxygen zone. They reproduce there with bioluminescence to attract each other and find mates. And they are the only octopus that are able to take care of their eggs on the water column. Really amazing animals. They were beautiful. Yeah, it's a treat for sure to finish the dive last night. Was amazing, and also you can see the skills trying to catch animals on the way up. They were telling me that's one of the. I mean, it's not the same to collect animals being with the ROV at the bottom, like stable, but it's another thing to on the way up. So it's a way that it's one of the things that they do to improve their skills. Hola, ven. ¿Cómo estás? ¿Todo bien? Okay. We are at waypoint 3. We are not at waypoint 4. I just made the mark
Okay.
by mistake. So we are finishing this heat flow measurement, and we will be moving towards.
Waypoint 4.
Yeah.
With in a transect or
with
just
no, with a transit.
okay.
Yeah. This was planned. We just because this was.
Yeah.
Forgot the lasers.
And then
So, yeah.
did we do any push cores yet?
Not yet. I think because we were doing transits.
Yeah.
But probably
That
if you
one.
can do
Yeah.
that after waypoint
Just
4,
put it
that
for
would be
me.
nice. Setting the pin ones in a specific place or
It's like it says by waypoint 6 things are gonna get rocky.
okay.
So I just want to make sure that
All right. Yeah, I will put them here.
I'm not.
So right now I will pass this seat where I'm sitting on to Diva Amon. Dr. Diva Amon is from she's as an NGO concerning about marine conservation, and she has a lot of knowledge of these abyssal plains and overall about the deep sea. So I will leave you with her. It was a pleasure. Good morning. En este momento voy a ceder el espacio a la doctora Diva Amon. Ella es de Trinidad y Tobago, con muchísimo conocimiento sobre este tipo de ambientes, de hábitats de profundidad. Así que las y los dejo con ella. Muchísimas gracias. Buenos días.
Morning, everyone. Good morning. How you doing, Ben? I'm down. Ooh. Me too. Okay. Okay, good morning, everybody. My name is Dr. Diva Amon. I'm with the University of California, Santa Barbara, as well as Species and Tobago, and I am joined on this science shift by Dr. Miguel Semedo from the University of Porto in Portugal. Have you guys introduced yourselves this morning? Ben, you want to go first? I'm just going to pass it to the RV pilots.
Presently the pilot right now, and my co-pilot Adam Whitmore, and over there is our RMT, Marine Technician. Morning to everybody.
Great, thank you guys. So we are just at Caballito, well, a seamount so-called Caballito. It's not its official name, and no one has ever been here before. We're exploring this area, and we've started in an abyssal area at about 3,100 meters depth. And we are going to be transiting across that abyssal area and taking heat flow measurements as well as transects for faunal community analysis. About halfway through those. And we're going to continue those, and then we're going to actually get to the base of the seamount and begin climbing that slope of the seamount and see what we see. As always, looking for areas of low temperature and octopus, evidence of octopus, but also seeing what the environment and animals are like on the way.
So we did the last one transit at 0.2.
Okay, perfect.
They say that they included that in the other ones.
I mean, I have a mixture of them written down for our transects, but it's fine.
We check the currents, which looks okay. I just want you to have a flag. Yeah, it was just starting. Yeah, we talked.
We're gonna reset some stuff here again. The stream isn't gonna restart. It'll just buffer for a little bit. So, and
Okay. Whoa, look at that crazy thing. Okay, so we have just reset our system, and if you're just joining us, are about 3,100 meters down on the seafloor. We are at a site somewhat known as Caballito. This is a seamount which no one has been to before. So we are currently in the process of, or I should say, our plan for the dive is to run from the abyssal plain where we are now at the base all the way up the edge to the summit of this feature. And along the way, where you can see us now, we are going to be doing some transects for megafauna community analyses, as well as taking heat flow measurements and push cores while we're on the soft sediment. As we're moving along, a lot of the soft sediment dwelling animals that we've seen during this expedition so far, as well as some new ones. Lots of urchins, like you can see on screen now, sea cucumbers, worms that We have not, to the question in the chat, we have not seen any beautiful nudibranchs. Nudibranchs aren't that common in the deep sea, I'd say. At least I don't recall seeing many on the expeditions that last night, though, we did, or yesterday, we did collect another type of mollusk, a beautiful velutinid gastropod, which kind of like masks itself as a nudibranch, kind of, but is actually a very family of snail. So also along the seafloor here, we can see a lot of these xenophyrophores. These are sediment looking like animals. They don't even look like animals, I guess. They are these ruffles in the sediment. And these are single-celled foraminifera that form these giant tests, and they can be reticulate, or we can also see some spatangoid sea urchins like this one here. These are irregular urchins, so rather than the regular ones, we see the circular ones that are what kind of comes to mind when you think sea urchin.
Anywhere.
Yeah, whenever you feel like you've going forward, we'll stop and we'll do the heat flow and the push cores, please. Sure, if that works for you guys. But then aren't you holding the heat flow, or would it be the other arm? Doesn't, okay.
I'll let you plan first.
And then boom. Great. So we're going to be here for about 10 to 15 minutes. And so we're going to take a look at what we see around us. Pardon? Oh, yeah. Oh, yeah. Great. And we're also going to take some 8 push cores so we can get some samples of the seafloor.
That'll be filled up here, right?
There should be nothing in the push cores.
outside first, put outside and inside. Just touch it.
Yeah, this wouldn't be a very bad operation. Right?
Why?
Now we're using those jaws, those particular jaws, because of the squeezers.
Mm-hmm.
Like if they had the regular jaws, it would be so much easier.
Which other jaws do you use? Oh, like the soft one.
No, it's more parallel.
Okay,
This one's
like this, yeah.
That's what makes this exercise a little bit more challenging. Like that.
So, yeah,
Like, we need it for the squeezer.
yeah.
Actually, this is the first time we've done cores.
Ooh, exciting. Not for you guys, but.
Adam, also known as the hammer, is now on.
Is that on or off? Oh, is that behind?
Looks like it's on.
Oh, it's forward. That's
So Ben, one of the ROV pilots sitting next to me, was just remarking that the manipulator arms that the ROV has, the two, you can customize them, so change the hands, if you will, the claws, if you will. And so this time they have on this manipulator arm you can see here has a very specific end that they're being used. And we have chosen that one because of the squeezer samples that we need to collect. So those are a very specific type of water collection that happens with this almost like syringe-like type instrument. And it needs this sort of rounded claw. But actually, normally, if we were doing push cores, it would be far easier just to use the manipulators that were not rounded and instead were sort of flat, if that makes sense. More like a tweezers.
Expertly maneuvered by the hammer.
Why do you have the nickname the Hammer? Do I want to ask that?
I'll let him answer that.
Wow, it is very sticky. Look at that stickiness. Are these the first samples? Are these the first samples? Wow, that color change is crazy.
right, folks. Audio might come across a little different, but I think we've managed to track down the choppiness we were getting. You might just get a little more fuzz in the background. You get to hear the lovely ambience of our mission control room AC. Although we're very, very grateful for the AC. Now is probably a good time for anyone who might have just tuned in. You are watching live from about 3,150 meters, just shy, below the surface of the Pacific Ocean off Costa Rica. And we're currently taking some sediment samples here. As you can see, they pull that tube up and it's got the dirt stuck inside of it. And we get all the nice little layers there. And then that sediment will actually be used in many different processes. Everything from looking at it under a microscope to look at its composition and the structures within it, to looking at the microbiomes that are contained within the sediment itself, the microbes that live in it. And I'm gonna hand it on over to Steph here for a little bit in the data logger seat, and I'll be back later. But we're gonna keep diving.
So with regard to the question about how long we're going to be doing these push cores, push cores can take anywhere from sort of 20 minutes to an hour. Yeah. And so we're only about halfway through. So certainly I would say the push cores would take us past the hour. And then after that, we're going to do a heat flow measurement, which should take another sort of 10 to 15 minutes. I know the heat flow measurement's happening now, duh. So at this site, we've probably got sort of 20 to 30 minutes, and then we're going to be on the move to waypoint 5. Yes. So if you're looking for a time to nip away for sort of half an hour, this is it.
When the core Tuesday coming on.
Yeah, I saw that. Still gives a 7. Hmm? The top came off, so there's no core. So what's next? How you doing, Mom? No, well, sure, your well-being, but also the heat flow's well-being.
Yeah, that's great. It's relatively low, consistent with overall.
What we thought. That is some really sticky sediment. Yeah. Means even sticking to the outside. Okay, this is our last push core, and then we're gonna, when we get the okay from the heat flow team in the room, we will withdraw the heat flow probe, and then we're gonna mosey on to waypoint 5. Great. Is your management finished?
I didn't start it, so
Oh.
yeah, I can't give the everything's jostling.
Oh.
So
I thought it was happening now.
Oh, I see. Yeah.
that's 29 minutes.
Great. So I was mistaken. I had thought because the temperature, the heat flow probe was in the sediment already, we were already in the process of doing the heat flow measurement, but that's not the case. So we are going to begin that shortly. And we will have another sort of 10 to 15 minutes here because we cannot be moving in the way that we are right now. Even though it would be very small movements, those vibrations and so on do affect the heat flow measurement. So yes, once we've put the lids back onto the push cores, we will proceed with the heat flow measurement. No, it was also just for my knowledge.
Yeah, typically it's
Bless you. I've asked the geologist, one of the geologists, Steph. So I'm waiting. So I have I'm not a geologist, I'm a biologist, but I have asked one of our geologists on board, Maria, from the University of Costa Rica. And she has said that these cores that we're collecting here, they're going to be used to study essentially time through the ages with by looking at the depth through the depth of these sediments. And she said that with every centimeter as we move down the core, we can potentially be looking at thousands to millions of years. However, we're not able to say anything more concise than that because she does need to look at the cores themselves before making a judgment call. So when these come back on board later, she is going to be sectioning them and then making slides and doing a bunch of other analyses back in the lab when she gets back to the University of Costa Rica. And there she's going to be able to build up this picture of the seafloor in this area of Costa Rica and what is going on there. So as we're doing the heat flow measurement, this is going to be a great time. I'm going to be handing over to Dr. Rachel Lauer from the University of Calgary, who is one of the two people on board responsible for running the heat flow probe and taking those measurements and trying to essentially use those measurements to guide where we are sampling. Ready? Great. Yeah. Do you want to just come and talk about heat flow?
Sure. Or you can. Go ahead.
I mean, I feel like for everybody's sake. Let me just tell you where we are.
Oh, yeah.
So we're currently waypoint 4 doing the heat flow. We did the push flows already. After this, you're going to drive to waypoint 5. You're not doing a transect between here and waypoint 5.
Okay, so we're just
You're
getting
doing one from waypoint 5 to waypoint 6. When you do them, here are the settings.
Got it.
Transects, 1 meter altitude, lasers on, no zoom, tilt just above tray, speed
21 meters per second.
And yeah,
That's it.
that's about it.
All right.
Alrighty, thank you everyone. Until tomorrow, I'm going to hand you to Rachel, who can tell you all about what we're doing right now. Thanks, guys. Leaving you in the throes of excitement.
I know, this is very, very exciting. Yes. For two of us.
Oh, by the way, I totally had an accident, so this is now.
Oh, don't worry about it. Yeah.
I'm in the electrical.
I love this. I love these caps, lasers.
That was, I think Bia wrote that on.
All right. Good morning, everybody. This is Rachel Lauer from the University of Calgary. I'm here with soon-to-be Dr. Rob Perrin, also from the University of Calgary. And we are in the process of conducting a heat flow measurement here on the abyssal plain during our approach to a seamount that was referred to as Caballito in at least one publication, so that's what we're calling it. This transect of heat flow is set up to try and estimate or understand the flow either into or out of this seamount. Actually, it may not be a seamount. That depends on whether or not it rises 1,000 meters above the sediment. So for now, I'll just call it an outcrop. So the idea is that this entire region... Oh, you stole a pen. Okay. What is it called? Oh, reticulate morphology. Xenophytophore?
Yeah.
Okay, I think
Single-celled foraminifera makes giant shells.
Oh my gosh. Single-celled foraminifera that makes giant shells. I hope I didn't screw that up. All right, good. Yeah, so this region and why we're doing additional heat flow here, our interest started way back at the beginning of the century. In 2001 and 2002, there were 2 heat flow mapping expeditions conducted over the, at that point, 10 known seamounts in this region. Those were generally aligned along transects that followed previous seismic lines, so that we would know the thickness of the sediment and allow us to use a 3-meter probe over the side of the ship to plunge into the sediments, measure the frictional heating and decay. From that, we get a geothermal gradient. After about 7 minutes, when that has equilibrated, we fire a calibrated heat pulse that puts a known amount of energy along the thermistor string, and the decay of that gives us the thermal conductivity. Those two multiplied together give you heat flow. And putting that in the context of the entire plate, as plates are formed at mid-ocean ridges, they're obviously very hot. They're molten material that cools very quickly. And a lot of these seamounts and outcrops are basically little volcanoes that are now extinct that formed at that ridge axis. As they move away from the axis, they become sedimented and cool. And that cooling rate is well understood using also models to understand the cooling rate and what we would expect the heat flow to be as a function of the age of the crust. In this particular area, the crust is around 20 to 23 million years old. And given that age, the heat flow should be about 110 milliwatts per square meter. Oh, there's a little guy poking out. Brittle star? Yep. It's a little brittle star hiding in a hole. What we see here, instead of the 110 milliwatts per square meter that should be coming out, is about 30 milliwatts per square meter. So 70% of the lithospheric heat in this portion of the crust is missing. And back in 2001, 2002, in the resulting papers that came out from those studies, it was suggested that this process of hydrothermal circulation was responsible for removing that heat and washing it out of the seafloor. And in order for that much heat to be missing, requires a very large crustal permeability. And this is sort of the holy grail of hydrogeology, is being able to estimate permeability, because it controls how fast fluids move through the crust, how long they hang out in the crust, and ultimately how much heat they can transport out of the crust. And this process of hydrothermal circulation happens when cold, dense bottom water sinks into one seamount that is permeable, makes its way into the crust below that seamount where it circulates, and ultimately makes its way to an exit point. Again, it can't exit across the seafloor because this entire region has been sedimented. In this particular spot, I think we have between, well, depending on where we are in this region, 250 to 450 meters of sediment. So there's almost no flow through those sediments. This requires that exchange between the ocean and the crust to happen at these seamounts or outcrops. So this process is called a hydrothermal siphon, where you have flow in one outcrop and out the other. And what we found on the past two dives on yesterday's outcrop, yet unnamed, but unfortunately we've been calling it the unnamed outcrop east of Caballito. We'll come up with something better. That outcrop had elevated heat flow, which is a sign that it was a discharge site. And fortunately, on subsequent dives, after we did the initial heat flow measurements, we were able to locate venting. And the temperature of the venting fluid before it mixed with seawater was up to just under 7 degrees, maybe as high as 7 degrees Celsius. And we can see that the temperature on the bottom here is about 1.85 degrees Celsius. Okay, so significantly above background. And that tells us that somewhere else that fluid is going in. It's warming up through its travels in the ocean crust. And when it comes out, it is transporting some of that heat with it. And in the case of that seamount, the unnamed seamount east of here, it was also hosting a community of brooding octopus near, or in the southern portion of that outcrop. So similar to what we found on Dorado, not as warm and not as prolific in terms of the numbers of octopus, brooding octopus, but we, of course, didn't explore the outcrop in the same, with as much time and bottom time as we had in previous expeditions at Dorado. Trying to get as much done here as possible in the short time that we have. And that includes identifying or exploring new outcrops where we have never been. And the one we're heading towards on this transect is one of those outcrops.
Rachel, I just checked the bathymetry, and
Yeah.
our lowest points, our abysmal plane is 3160 roughly,
Cool.
and the peak
is around 2790. So over 300 meters.
Okay. Over, you mean the
Top of the outcrop.
Okay, awesome. Yes, so this is definitely not a seamount. It is an outcrop. It's about 300 meters above the seafloor.
370 or so.
370 meters? Cool. So this one we had, it was visible in previous data sets that were collected, the multi-beam bathymetry, but we did a couple passes over it in the past week to increase that resolution and identify where the steep sides were that might be potential hosts for octopus, as well as the one east of here. So we tightened up that bathymetry and got a much better picture of what's going on here prior to planning today's dive and the dives we did on the eastern outcrop. So we're doing our fourth heat flow measurement now. And as I mentioned previously, the heat flow in this area should be about 110 milliwatts per square meter based on just the age of the crust. This whole region, which is roughly the size of Connecticut, it's about 14,500 square kilometers, is missing 70% of that heat. And the average heat flow is about 30 milliwatts per square meter. All right, so we just finished with our fourth heat flow measurement.
Into the box.
Oh, you got it in the back.
So the lance that we just removed is used for two reasons in these measurements. We use it first to test whether or not we have a full meter that we can safely insert the probe into that soft sediment to ensure that we don't damage the probe. And it also provides a little bit of stability while we're conducting that measurement so that we can eliminate, ideally, all the noise associated with SuBastian, keeping it still while we do those measurements.
Are you ready?
Yep. All right. So we're going to make our way to the 5th waypoint. Oh. Got another tripod. Is it? Yeah. Not sure, actually. Not a tripod.
Bridge, ROV, that's an updated waypoint for you.
Update waypoint, thank you.
Thanks. Yeah, I'm trying to keep it straight on.
So you can see in the map in the top left corner that we are heading towards the base of this outcrop, Caballito outcrop. Look, we'll probably get one, maybe two more heat flow measurements before we start ascending, and it's a little bit too rocky for us to make those measurements safely without injuring the probe. So far, those measurements indicate relatively cool conditions as we approach. We haven't modeled to actually come up with a specific number, but they are within the range we would expect for this region. What's that? Yeah, these are the first measurements ever using heat flow. We have no seismic for this region. The bathymetric mapping that was conducted from this ship was the first refined look at this structure at all. And we are, on this dive, going to be the first people to take a look at this structure. So this is really exciting. And the heat flow measurements are exciting for, certainly for me and student Rob. They help us understand sort of the hydrogeology of this system, not just the plumbing underneath, but whether or not these outcrops are recharging or discharging. If in fact this is a cold outcrop, it suggests that this structure is pulling water in and potentially exchanging with the outcrop to the east that we spent 2 dives on previously, where we found the newly identified octopus nursery, as well as sites of warm venting. Okay, so we're almost at the 5th waypoint where we'll be doing another heat flow measurement. We're in the spot. Cool. It's always nice to choose a heat flow position where there's a little bit of fauna to look at. All right, so this is Excalibur, again, testing both to make sure that the sediment is deep enough for us to make a heat flow measurement. Needs to be a meter. And then also this provides a bit of an anchor to reduce vibration from the submarine while we conduct the heat flow measurement. Cool. You guys ready, or?
We're always ready.
You're always ready. All right, Rob, give him the go when you're ready.
Yeah, I'm recording.
All right, good to go. There you go. That is a great. Okay, so what is this again? This is a sea anemone and a crab? Yeah. A what? Okay, it's a crab. Yeah.
Is that getting you your 5th sensor?
Is it? Are we all the way in?
Yeah, the 5th is like close, like it's borderline. There's a little rise.
Okay.
Are you all the way down then?
All the way down.
Wait. Maybe. I don't know if you want to try it further down.
How far into it are we?
We're 80 seconds in. We could start a new one if you want to do that.
If it's did it bump up at all, or?
A little bit, like a tiny bit.
Okay. If we can we do another?
Further?
Yeah,
I
just
still want to reach sample.
yeah,
Yeah.
or maybe
So you spell it
Oh, pagoda.
Pagode, yeah.
Okay.
There are two types of perfect.
It's not all the way down. It's all the way
Okay. We'll just go with it. Okay. All right. So this is a pagoda crab with an anemone taking a ride. Obviously, some kind of symbiotic relationship going on there. Wow, beautiful. What is it? Eredicanthus? Luckily, I'm surrounded by biologists who actually know how to identify all these things. It does look like a truffula tree. A truffula tree. Yeah, from the Lorax. As mentioned in the chat, this plate, this portion of the Pacific plate is called the Cocos Plate, and it is subducting beneath Costa Rica at what is called the Middle America Trench. That portion, that right where it's subducting is estimated to be 24 million years old, so where we are now is younger than that. And the sediment ranges from between 150 to 450 meters along the trench. It's a little bit rougher to the south, so the sediment thickness is reduced a little bit. Where we're subducting underneath Nicoya Peninsula, it's about 450 meters of sediment. Interesting point about the heat flow and how things are actually different at the plate boundary here. Some of this crust originates from the East Pacific Rise, and to the south, the crust is originating from the Coconasca Spreading Center. They are similar age and sutured or stuck together. And during subduction, there's lots of studies using the Nicoya Peninsula, which sits right above what we call the seismogenic zone. And that's where earthquakes can initiate. It's one of the few places in the world where we have land directly above that, where we can instrument using seismometers and other types of installations to monitor closely for seismicity. And the results of a study that was conducted in late 1990s, early 2000s found that on the cold side of the plate, earthquakes were happening deeper than on the warm side of the plate. So Costa Rica is a fabulous laboratory for thinking about seismicity. as well as obviously a great place to look at biodiversity and octopus. So that study, I think it was published in 2001 by N.D. Newman, suggested that the thermal state of the crust is responsible for determining where earthquakes happen at the plate interface. So in a previous life, I studied subduction zone hydrogeology. So thinking instead about how fluids move between those plates along faults in the upper crust, and how fluids and fluid pressures may play a role in when, where, and how earthquakes happen. Oh, he's on the move. Not enjoying the spotlight. He is dancing, yeah. He's tired. All that dancing has him a little tired. He's taking a break.
chance to look at those and stuff like that. Yeah, he posted up prior. Okay. I think it looked cool. Similar maybe. Yeah. Okay. Right.
Oh, you did. Awesome. That's beautiful. And that thing in the middle, what is that?
Okay, that's good for that.
Yeah. Oh, polyheat. Okay. All right. Done with heat flow measurement number 5. And we are doing a biotransect, correct? The next one. So for that, you know this.
You went to which one, 6?
Yeah. Apparently.
That was the end of 5.
This one, eh?
Yep, we're going to 6. Yep. So lasers are on and 1 meter. And 0.1 meters per second. guess no zoom.
It's on the ROV, see on the display it next to the wall. So right now it's
How are you?
yeah. So I think probably one more from that. And then I'll take up, yeah. Yeah, I don't know what the plan is. My transect's not numbers.
Oh yeah, go for it. I think what we switched maybe, this is now a bio transect.
Right.
And the parameters are up here. So 1 meter off deck, no zoom, lasers on, rate, speed. And we're going from 5 to 6 now. So this is, we're on this one, but we're heading to this, if we can. But honestly, the sediment should be thinning as we're getting closer.
Bridge, ROV, that's waypoint 3.
So just on this heat flow, we're doing this transect.
Yep.
Copy, thank you.
Doing another heat flow.
Yep, ideally, I mean, we'll see, we have 1, 2, 3, 4, 5, 6, 7, 8. We don't expect to get all of them, but as many as we can.
This heat flow and this heat flow the same thing? 'Cause it's like heat flow transect. heat flow
No,
transect.
it's confusing, because I think we did a laser, or a transect here instead of here, because it didn't happen here, so everything sort of shifted.
Okay.
This is, I had a very quick conversation with you about it. She said, next to the last one, no kind of this one, which means, it's a big confusion.
Sorry.
Yeah, no, it is, it's
So I transect now towards
this
this
one, towards 6.
6. Yep. Okay, we're gonna do
Where-
a heat flow there.
Yep.
And then are we doing a transect after that?
No, I think it's transect, not transect, transect, not transect, but I'll ask for you.
Okay.
I'll go ask you that. There she is.
Did I leave my mug in here?
No, I don't have it. Beth is missing her mug too.
Okay. Yeah, I was just wondering about the transect spreadsheet strip explain. I just want to be sure I'm right. So we're doing transect 3 right now. Then we're
doing 1, 2, yeah, so this week, 3, yeah.
So then we're doing heat flow.
Yeah,
At
and
1.5.
1.5.
No, we're heading to 6 now.
Sorry, 6, yes.
And then are we doing a transect between waypoint
No,
6 and 7?
you're gonna do one
Every
between
other.
7 and
Go
8.
to 7 and then do heat flow. Exactly. Okay.
Thank you. That's, I thought that was right, but I just glad she
Yes.
clarified.
Sounds good.
Oh, interesting.
What have you been doing when you haven't had to do transit? Just zooming around, like
Just going faster and
Yeah.
zooming. Yeah.
speeding up a bit.
Yep, exactly.
Sounds good.
Talking about heat flow and seduction.
Yeah.
It's like staying in my safe place.
Just go from a really good explanation of that to like, well,
Sticking it in.
it means something.
I'll leave you with it right before I'll give you an explanation. You don't have to worry about it.
It's really
Yep. Yeah, so during these transects, we leave the lasers on because those provide us with a quantitative estimate of the size or scale of some of the things that we're observing. And we don't zoom because we want to keep a wide swath visible for identifying creatures along the way and make our observations. Ooh. For those just joining, I am Rachel Lauer from the University of Calgary, joined by Wendelin Matamoros Calderon from the University of Costa Rica, and Robert Perrin, soon to be Dr. Robert Perrin, also from the University of Calgary. And we are heading towards our 6th location for heat flow transect. We are gaining elevation as we approach Caballito, another seamount west of our location for a dive yesterday. And just to catch those up who just got here, yesterday's dive was on a discharging seamount where we found venting in many locations as well as an octopus nursery. That seamount had elevated heat flow, and We are now diving on a seamount about 2.5 kilometers west of yesterday's location. We're currently about maybe 600 meters from what we expect to be the rocky outcrop. We need the sediment cover to actually conduct our heat flow measurements. So we started out in what is the abyssal plain. And in between those measurements, we are looking at the benthic fauna to conduct diversity studies. So we stay about a meter off the sediment and move at a constant rate with the lasers on, again, to provide some quantitative estimates of the size of some of the things that we're observing at the seafloor. Yeah, it's crazy. Oh, one of my favorite things we've seen is a tripod fish. We could, yeah, let's do that. We're in our location for next heat flow measurement. Hopefully this guy sticks around so we have something to look at while we're doing that. Okay, so it looks like we have soft sediment here and plenty to make our heat flow measurement.
The thing is too high off.
Mm-hmm.
We're starting to go up a little bit.
Yep.
So we might just pick up and try to
The other way.
sideways into it. Yeah.
Awesome. Okay, we're going to reposition a bit to ensure that we have all of our thermistors in the sediment before we start our measurement. We're on a bit of a slope here. He's still there. I think he moved a little bit. Very good. I'll just talk a little bit while the dust settles. So we are on the site of our 6th heat flow measurement, heading towards Caballito. And this pair of outcrops that we've been focused on for the past couple of dives are right at the edge of this thermal transition where we have a large region about the size of Connecticut that is missing 70% of the heat that's been washed out.
This is actually much better. You can see you're like level with.
Oh, you can see the heat flow guy?
It's level with the.
Okay. Okay, so we can see in the camera that matters, the one that's on, sorry, the camera that we need to ensure that the heat flow probe is okay. Yep, awesome. Okay, Rob, are you all set?
Yes. I haven't probed it yet with the caliper. That's there.
I think I mean, it's in front versus over here anyway, so
I'm happy if you're happy.
I think I'm happy.
We'll watch it here, and if we see anything.
If it does any,
yeah.
yep,
Right back.
okay.
Right.
You ready?
Start recording now.
Yep.
Here we go. Okay. I'm recording.
Oh, yeah.
No, I think this will be the last one.
Okay. Awesome.
Turn sideways and put this too far off. So that's right. Those temperatures. This will turn sideways. Oh, the transect, I think maybe one more. I don't have Excalibur in here right now. And the delta's a little bit bigger just because you're waiting for me to get closer.
See now. Enjoy your lunch. The chattering tree.
Yep. Yeah, thank you. I forgot we didn't put him in this time. We moved because there's nothing to see. We're in a sandstorm right now.
We want to get this done before we have to go back and forth.
Exactly. Exactly. All right, so it's been a pleasure chatting with you for the past hour. I am going to hand over to Emily Cowell from Temple University, soon to be Dr. Emily Cowell.
That's the least impressive person to be. So much better.
Good, how are you? Nice. Surviving.
She was like, how's your day? I was like, she was busy. I was like, I had one of the deck really huge. And I did this and my transects me too. I was like, I couldn't stay back. Done.
That's been a few of my days.
Did you get all three of those? Steps, did you get that already? Oh, still
I'm
there.
on this one. I don't have the tab. How long does it
Oh,
take? So it sends a heat pulse into the setting?
And then the decay from that sensors
still doing the transects in between?
We're not doing a transect between this one and next one. We're just speeding up and we can zoom in on things. And then after the next one, we're doing a transect. Yeah, it's every other.
So I don't have to make my way back to the actual spot.
No. Yeah, after this, we can just go straight to 7, and then we do a transect between 7 and 8. And I think that's the last transect.
Okay.
Yeah. Are those the oil pressures?
Yeah, those are compensators.
Cool.
5.4.
Oh, just ruined it. Now I don't know what I'm doing. Just kidding.
That's no idea.
Now that data is useless to me.
Exactly. Wrong. So for anyone that's just tuning in, the view right now on the science camera, I think, is probably the just the pressures of the. That one? Oh, nice. That's good then. So we're doing a heat flow measurement. So that'll take about 15 minutes. We're trying to measure the heat flow through the sediment of all of the sites, if possible, that we're studying, to see if we can see evidence of them being higher or lower heat flow than kind of expected, which would suggest whether it's a site that might be discharging fluid or recharging fluid into the sediment. So the heat flow probe sits in the sediment for about 7 minutes, and then it sends a calibrated heat pulse into the sediment and watches the decay of that for another 7 minutes, and then that'll be the whole measurement. And then after that, we will head off across the sediment again towards our next waypoint. We've been doing some biology transects between some of the waypoints where we have the 10 centimeter lasers on, and we're going at a very constant speed, and we've angled the camera at a very specific angle. We don't sort of move the camera around or zoom or anything. And that just allows us to, when we're back on shore, do some biological counts, look at densities of organisms across these transects. We'll not be doing that between this waypoint and the next one. We'll be able to have a bit more freedom, go a bit faster, and move the camera around and zoom in on some things. But once we get to the next waypoint, do the next heat flow probe, then we'll do our final transect.
Whenever you're like digging a flower, you just make sure it's fully covered. That's what we do with our trash. I joke, but we've all done it. That's fine. There's something that's fine.
So this is a 16-hour dive for anyone who's just tuning in. It's a return dive to a site that had never been explored before. That's being called the Caballito Seamount. We have done some dives on an unnamed outcrop east of this Caballito Seamount and seen some evidence of fluid discharge, some octopus, kind of like a less dense octopus aggregation than we saw at Dorado, but a similar setup. So it'll be interesting to see what the situation is at this Caballito Seamount. I think once we get past waypoint 6, the terrain's going to become quite a bit steeper, so we'll probably not be doing any heat flow measurements much past that point. So the heat flow has to go into this soft sediment, because the probe is about 1 meter long, and it needs to go all the way through into the sediment, so all of the thermistors on the probe are in the sediment. Then once we're on that steeper terrain, we're going to switch our focus to just exploring the environment, looking for signs of venting. So previously we've seen signs of venting as brooding octopus. We've seen fluid discharge that looks kind of like shimmering waters coming out of cracks and holes in the rock. And also areas where the rock looks darker than the surrounding areas because it's actually been cleared of the sediment. The fluid flow has wiped the sediment away from that rock, so it looks darker. So it looks like we should be aiming to get to the summit by about 4 p.m. local time. It's 11 a.m. just after local time now. If we find any venting or octopus aggregations, we'll try and sample them, deploy a temperature logger and a marker so that we know the locations of them for if we come back to this site in the future. And we'll try and collect a couple of octopus specimens from this site and see if there's any other interesting fauna around that we've seen during the dive, and mark their locations as well. Looks like the little brittle stars that have buried themselves. I don't know what the thing is above. These little arms you see sticking out of here are probably brittle stars. Often they'll bury themselves kind of in the sediment and stick their little spiny arms out. Little unidentified ball of sludge. It's a thing. Oh, and it's off. Yeah, is that where the ROV landed down before?
No, that's been there forever. Call that uplifting.
I see.
Yeah, I assume that's probably what
Yeah, it's really soft sediment around here. Seems like it has more of this kind of white sedimented layer underneath this top brown layer of sediment. That stuff's been really interesting when we've got it onto the ship. It turns into just sort of white ooze, even though it looks fairly solid on camera. Yeah, like this kind of stuff that looks a bit like more like rock. When you actually get it to the surface, it's just like sludge. It's a shame the tripod fish swam away. We could have had a really good view of him. I'll lodge a complaint.
That's fine. That was something that was just a shadow. Seriousness though, that's probably not from the ROV.
Yes. Do you do something that sends the
No, the pulse. We wait 7 minutes after the pulse and then stop measurements.
Yeah, I just mean, does it send automatically, or do you have to do something?
We have it to set automatically. You can also do it manually.
Yeah.
But we have it set for 7 minutes after. Well, it's actually 8 minutes after the
Yeah. From the readings you get, are you able to see sort of straight away what the trend is looking like, or do you have to?
Yeah,
Look
because
at it
we do.
together and-
You have to do a bottom water correction.
Right.
Apply that to the data, and then we always try and guess, and we're almost always wrong.
Yeah.
So, yeah, it's better not to. It's tiny little corrections that matter.
Right.
So, yeah.
Okay. So in a second, the heat flow probe will be sending a heat pulse down into the sediment.
Oh, it did already.
Oh, it just did by the sounds of things. And then we wait for 7 more minutes to watch the decay in the heat flow of the pulse that was sent. And then after that, we're good to go and move on to the next waypoint. And then we'll do another heat flow again.
Hey, we're all good.
Cover the probe.
No, nice. We need a minute until the end of the whole thing.
That's it's okay.
So,
Yeah, that's good.
really?
Yeah.
Oh, I thought she said that was all good.
Huh?
I thought she said she was all done.
Yeah, she did. Yeah, I was getting mixed up.
Oh, I'm confused.
I'm just confusing you. It's fine.
Just gonna fly my ROV. Alright.
Right. So now that that's done, we can set off towards the waypoint 7. So we're not doing a biology transact between these two waypoints, so we're free to go at a bit of a higher speed. We can also zoom in on anything that's of interest. Oh, there's a tripod fish in the background. They're always of interest to me. These are so cool. We've been seeing quite a lot of these on these dives. They're just such interesting fish. They have these big projections coming from their fins that allow them to kind of rest on their fins. So strange.
Alright, it's gonna get funny since we're full of lights, so I apologize.
Fair enough. These tripod fish have been a personal favourite of mine. Certainly up there amongst the favourites from these dives. They're just really otherworldly. It's so cool.
Back to work.
Yes, I suppose.
I think we'll put some behind us.
Hey, on the bridge, we got an updated waypoint for you.
It's so weird listening to that. I have to keep working so much.
So it's still looking really sedimented as we move up in the direction of the slope. Well, up in the direction of the steeper slopes. So hopefully we'll get a couple more, or at least one more heat probe measurement in. So once we get to this, what's likely to be a kind of rockier slope, it's not going to be possible to do the heat flow measurements anymore. So we're making use of all these sedimented areas. Looks like a sea cucumber.
Bring it up, get down to M2, like 65.
There's been lots of excitement about the sea cucumbers on our cruise so far, from certainly a couple of the members of our team who are interested in looking at the microbiomes in the guts of the sea cucumber.
Primarily the hindgut, where we believe there's an end of symbiote.
Oh, cool. So just in case you couldn't hear that, that was,
That's okay for now.
yeah, they're interested in primarily the hindgut of the sea cucumbers, to see if there might be an endosymbiont associated with them. But we've had a lot of collections of the sea cucumbers so far. We've got a nice variety.
I'll write down this like shit on a fish. Yeah.
A nice close-up view of this guy. Whoosh. Looks like we've got some larger rocks appearing in the background. Some corals and sponges may be associated with them. We'll come up and explore that a little bit. Another fish down the bottom there too. So it's impressive to see these really large coral fans starting to appear here. What we were noticing on the sides of the steep sides of the rocky seamounts that often you didn't see as many of the large corals, or if you did, there was quite a lot of broken coral around, probably from being damaged by falling rocks. But on these flatter areas, you can see they can get up to these really impressive sizes. They have very slow growth rates, so these are very old organisms.
We know how long something of this size might be.
How old is something like that? Yeah, it's probably several hundred years old, most likely. Certainly some of these really big corals are very, very old. So the growth rates of a lot of organisms on the sort of these background areas in the deep sea are usually very slow. Got cold temperatures and lower food supply, so slow metabolisms. Often on the on things like hydrothermal vents and methane seeps, it's a bit faster because you've got heat input and higher levels of food supply. Certainly if you've got chemosynthetic symbionts.
Just rolls off your tongue.
Chemosynthetic symbiont. Yeah, sure. So when we had the little zoom in there of the corals, you see the kind of star-shaped structures. Each of those is a polyp, and each of those polyps is its own individual animal. So corals aren't plants like we might kind of expect. They're animals. So in shallow water, they usually have an associated photosynthetic symbiont called zooxanthellae, and they use those symbionts. The symbionts photosynthesize, and they kind of use they use that for a food supply. In the deep sea, we tend to see that corals, rather than having this, they don't have the zooxanthellae because you don't have the light here for photosynthesis. So instead, a lot of them are sort of maybe filter feeding organisms, although there is some suggestion that some deep sea corals, especially around areas like methane seeps, might have chemosynthetic symbionts. But yeah, the entire structure is a colonial organism made up of lots of individual animals. So each of those star shapes there is a polyp that's an individual animal. Cool. Thank you. Looks like a pneumaticosinid shrimp there on the ground. We've collected one of those yesterday. I'm excited to have a look at that and see if I can ID it. They've got these, just that shrimp there. But we've seen lots of those on the dives. They've got these really, really long walking legs. It's okay. We saw a load of them yesterday. We got some good footage. Thanks. I'm always getting too excited about them. Doing lots of zooms. Oh, you want to go to... Can we actually spin around to the right a little bit and have a look back at that rock? It looked like there was a black coral that Wendy's interested in.
It's not really like
Mm-hmm.
it's frustrating.
What I'll do is I'll have to sit down and draw that out from underneath us.
Uh-huh. Yeah, we've got a lot of, from doing the heat flow measurements and from this really sedimented environment.
So it's,
I mean, we can have a look at it now. I think it's kind of underneath. Yeah. We've got a lot of mud on the ROV. So there, as we sort of reverse the mud, it creates a cloud around us.
where exactly on the rocket do you want to look?
Is it? Where's the stick?
I see something right
The
there.
pointy stick.
Right of that rock.
Is it this? Yeah. That one there. Cool. So it looks like there's a black coral, and that kind of one that almost looks like a big fat feather. Top right of this rock. So we're going to have a look at that. Are you just wanting a good look at it and zoom in things? Zoom over polyps. Cool, okay. Do you want the lasers on as well? Yeah.
I'm gonna land out right here in this corner, and I'm gonna dig it nice.
Great, thanks.
Being on that rock's gonna get difficult.
Sure. What's this bit called that you grab onto the rock with?
We
got a name?
call them rock and socket. We usually have a second one on the starboard side, but we had to remove it for the heat flow.
Oh, I see.
Yeah. There are side rails, but we call them.
Does it make it difficult only having one?
No, it limits it to the point where our options are not as plentiful.
Right. Okay.
And in situations like this, we wouldn't usually do two of them. Sometimes we would, but for this kind of
Yeah.
so far ahead, it makes sense.
Nice. Yeah, we just wanted to zoom as close as possible of the polyps, kind of a glamour shot of it for a little bit, and then also see if we can have a zoom with the lasers in a second. Thank you. In a minute, I think. Yeah.
What is that thing?
The little amphipods, maybe. Does it look like a tiny little snail?
Alright, there we go.
Emily, what is that thing?
Well, I think a lot of them look like possibly amphipods, but we're thinking that looks like a tiny snail on the bottom right as well.
Tiny snail on the left.
Amphipods plus snail, okay.
Yeah, I could be wrong about that, but
I think you're right.
maybe amphipods plus snail.
It's
It's a snail. So tiny. Yeah, and then
you
if
go ahead and pause right there. We'll be getting back to going in just a second, but taking a couple
Yeah.
pictures.
and then if we could
Thank you.
if we could get the lasers on just on maybe on the rock on the bottom,
Yep.
that would be great as well. Cool. Can they be pointed towards the rock down here? Thank you. Does that work for you? Nice. Great. Yeah, for anyone wondering, those two green dots are 10 centimeters apart, so it allows us to get an idea of the size of the organism. Cool. Are we good to go? You want to do anything else? Good to go? Okay. Yeah, that's great. Thank you. We could probably turn the lasers off again now. Yeah.
Turn back
Thanks.
actually and just turn off for a second. I can see something. So, it's just not even- okay. Make sure they don't hurt it.
So for anyone that's just tuning in, this is a return dive, a 16-hour return dive to a seamount
We're gonna do it again so you can
off the
Awesome. Thank you.
Pacific coast of Costa Rica. So it's called Caballito Seamount, or it's been sort of referred to as Caballito Seamount. So we've been conducting some heat flow measurements along the abyssal plain to the east of the seamount. We're moving in a west-southwesterly sort of line towards the seamount. We've done some sediment push cores. Between each of the heat flows, we've been doing some biology transects as well, where we have the lasers on and we go at a very constant speed. We don't move the camera and we don't do any zooms on any organisms. That allows us to do a really nice transect where we can count densities and of the organisms that we're seeing. It allows a more quantitative sample to be done.
So we will not be able to do any more heat flow.
Yeah. Okay. Our intention had been to do a couple more heat flow measurements if possible, but it's starting to get pretty rocky here, so I would suspect that that's not going to end up happening. We need very deep sediment to be able to place the heat flow probe, because it's about 1 meter long and it needs to go all the way into the sediment. Cool. So we're making our way towards our next waypoint, where we were intending to do a heat flow measurement. If that looks like it's not going to happen, then I think what we'll do once we get there is do our final transect. And then after that, we'll be firmly up onto the slope of the seamount, and we'll start investigating the seamount for evidence of fluid flow. So for anyone that's just tuning in, when we're looking for evidence of fluid flow, at previous sites we've seen brooding octopus as evidence, because they appear to, at the other sites, like to brood their eggs in the fluid flow. We're also looking for shimmering water, because the water is slightly different temperature and density than the ambient. And then finally, we're looking for areas where the rocks have been swept clean of this light-colored sediment, so they look much darker in comparison to the surrounding rock. And that would be where the fluid's been coming out and sweeping the sediment off the surface of the rocks. But we're almost at waypoint 7 now, so once we get there, we'll have a think about doing this next transect. Yeah, so you should be able to see there on the little insert that we're just coming up the edge of the seamount now. Hence why it's changed from that flat, muddy abyssal plain to the steeper, rocky environment.
Okay, so waypoint, there is some
Can ask
sediment area.
Rachel.
You want to give it a shot.
This is around waypoint 7. Do you want to try and do the heat flow?
I mean, we can poke a little bit, but it's starting to look a little bit,
Yeah.
yeah.
Okay.
dicey.
All right.
I mean, this looks like this area is quite sedimented right
Right
here.
here is where
Yeah,
try it.
give it a go.
Overside. Look,
See him maybe.
I want to spin 90 degrees.
Mm-hmm. So this is a relatively sedimented spot here. So we're going to set down and poke the sediment and see if it's a meter deep so that we can put the heat flow probe in. We have to be careful because the ROV is very powerful, so if you sort of press too hard you can bend the probes very easily if you're coming up against a rock or something that's buried in the sediment.
You can choose yourself to go in and say,
This is a very, very hot. It's gonna be messy. Hit me up, 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 hit me up. Hmm? Alright. Alright, should be somewhat stable. I like sitting on this.
Weird angle.
Yeah, so.
Okay. So we're going to give it a go and see if we can find a spot of sediment right here that is deep enough for the heat probe.
For this new probe here, I want to go like right in that area. We're definitely on an area like this right now.
Yeah.
So it's close to the I can go closer to us if we can. Yeah, try to get even closer. So it's more definitely the same kind of situation as working with the probe. Alright, go for it. It's a little bit hard, yeah. I wouldn't risk it.
Nope,
Okay.
let's bag it, thank you.
Yeah, you could just see that about halfway down there you started meeting quite a bit of resistance. We want to make sure we're not breaking any equipment. So
I'll come up and
I
get
think
some
in that
food
case,
instead of wet.
we're doing a transect between this spot and the next waypoint.
All right.
So the next thing we're going to do between this waypoint and the next, which is about 100 meters, is to do another biology transect. So that involves us setting the view of the ROV to be sort of standardized at each transect. So we'll be 1 meter above the sediment. We'll have the lasers on for scale. We won't be zooming the camera or adjusting it in any way. And the camera will be tilted to sort of just above where the tray of the ROV sits. And we'll have a constant speed, so we'll be going at 0.1 meters a second.
Alright, so I'm going to try to get us to what looks like the same height as where I know it's going to fly.
Mm-hmm. We're just doing the last transect.
It might be beneficial as well to bring the camera up more than normal, just because that's the angle we've seen of that.
Mm-hmm.
Sneak it through like this instead of flat.
Okay.
It's up to you.
I was just saying that, do we want to tilt the camera up anymore because we're now on a slope? Or do we want to keep it the same as tilted to just above the tray?
Yeah, so this is basically where I thought we would run out of the possibility of doing transects, just because it's going to get so difficult, and now that we're in rocks, we want to actually start exploring for venting.
Okay.
So if you're already setting up for it, that's fine.
Whatever you think.
And yeah, I guess for this next spot, we'll just do that. We'll go up to the next waypoint.
Okay, so do a transect.
Yeah, maybe. Because if we see sediment, we're going to want to stop to get a heat flow measurement.
Mm-hmm.
Yeah, so I think at this point, now that we're on a slope, like getting transects is over
Okay.
for the dive.
Sounds good.
And so we probably want to come away from the seafloor so that we have the ability to like pan around and
Great.
look for interesting things while we're following the waypoint. And if we see a spot that looks heavily sedimented, like sit down and poke to see if we can get one more measurement.
Cool. Sounds good.
Does that make sense?
Yeah.
Does that work for you, Chris?
That's right.
Okay.
That works for me.
Yeah. Now we're in exploration mode.
Sounds good.
Plus trying to get one more heat flow.
Okay, so we're keeping an eye
Funky
open.
transect.
Yeah, we're not doing a funky transect.
Alright, awesome.
Nice.
Okay.
Well, that works.
I'll send that as a message to everybody so they know what we're doing.
Okay, so there was 3 transects in total. Does that sound right? Something like that. This one, that one, and that one.
Yeah.
Yeah, great.
I thought we would maybe get 4. But
Okay.
that's fine because we didn't start at the right time.
Right.
Yeah, the 1, 2, 3. Yeah.
Okay. Great. Sounds good to me. Yep. In that case, a change of plans. We're not going to do the final transect that was on the dive plan just because of the slope here. It becomes quite difficult to get good transects. Now we're in these rocks. We want to keep an eye open a little bit more. We also want to keep an eye open for large sedimented areas to try and get a final heat flow measurement.
Costa Rican narwhal, right? Right, Jimbo?
Sorry, was it?
Keep an eye open for the Costa Rican narwhal.
Pacific novel.
Great Pacific narwhal, okay.
Get your names right.
If a narwhal swims past the camera right now, I will be so impressed.
If the narwhal.
Well, you get a paper out of it, that's for sure.
That's true.
I would stop the transect for that.
So it looks like we're seeing a few more of these sponges. Might be starting to see some more corals as we're coming up onto these slopes as well. So we'll keep our eyes open for those. We've got multiple cameras here in the ROV van, so it allows us to have slightly wider view than what you can see on the science camera. So we're also on the peripheries looking out for any areas of sediment where it looks like we might be able to do a final heat flow measurement. So we'll keep our eyes peeled for that too. Though it's looking very rocky as we come up here, so the chances of that might be quite low. Yeah, here we go. Loads of ophiuroids and sponges on this rock. Ophiuroids are the brittle stars, pink stars that you see there. Pretty large rock just out of nowhere.
Wow.
Some pretty big sponges as well. Some sea cucumbers, a variety of things on this rock here. Crinoids maybe even. Yeah. Yeah, so that was, for anyone interested in the geology side of things, that was a large pillow lava. That,
Put a marker down for us.
I think they just marked in you weren't wanting actual markers, you were just wanting markers. Oh, great. And this is a carbonate ledge here?
Yep,
Cool.
but not with the same crusty bit as we see at the other sites.
Right. So that looks more like it's actual carbonate, like carbonate rock?
No. It's still this
Ooziness?
ooze,
Right, there's the ooze.
but it doesn't have the black crusty ledge on top.
Right, I see.
One way to find out.
It's your destiny.
Chuck under a ledge. Looks just like a rock.
The discoloration is suspect.
I'd rather just mark it to see what it looks like, and then I can come back. Can you put in a mark here that we see this carbonate ledge? The problem is we don't have anywhere to put it. Because the bio box is in the front and full, until we put stuff out. And we don't have the rock box. And the bio boxes we need to save for animals.
So this ledge is made up of that same carbonate kind of ooze, this white material that we were talking about earlier. So it looks like hard rock right here, but when you actually sample it, it's very soft, and when it gets to the surface, it sort of disintegrates into
We're going to do at least one more dive here.
pretty much liquid.
See how this looks. And try to-
It looks like we're seeing more of these pillow lava too, coming up here. It's amazing how quickly the environment changes as soon as you're up on the side of the slope. And of course the organisms that you see along with it. Looks like some little polychaete were maybe there. So as I say, as we're coming up the side of here, we're investigating first off any rocks that might be of interest to collect later. So we've been putting some markers down for that kind of carbonate ledge and the start of these pillow basalts. We're also looking for areas that might indicate some fluid flow. So particularly in these spots, it would be dark rock areas where the sediment's been sort of blown off the surface of the rock, revealing these just very dark rocks underneath. If we did end up finding any areas of fluid outlet, we would take a temperature measurement with a high temperature heat probe, and we would potentially try and do some water samples as well. Ooh. Looks like some more crinoids in the background there, certainly.
Based on the geology, this better, that's better, this part better.
Pardon?
Finding any kind of showing water, you think that the geology aspect of this side is better, this side is better, or is it all pretty much?
I don't know exactly in terms of the geology. I think we've been mostly seeing it in kind of large cracks and crevices in the rocks. So often in kind of like troughs and things between rocks, but don't really know. I think it seems to have been.
I don't want to focus on here and like right around the corners, like.
Yeah. Yeah, I think it's been
Sorry, Tony, just
it's been a bit of variety.
Can you repeat?
Yeah, I'm just bringing myself to a stop now before this waypoint, just to let you guys get ahead of me again, so that you don't want to the rest of the
Yeah. Roger that, Tony. Thank you.
Okay, tell me we're moving forward, but we're also looking up at a higher clip, so it's going to be a little while for us to move forward.
Yeah.
Yeah, Tony, just to let you know, we're moving up in cold.
Yeah, copy, man. Thanks.
More of these large octocoral fans up the side of here too, and some sponges. Yeah, so
We are just going, yeah, we're looking for that over there, because it was cool, so sorry. We're looking for any kind of warm water or water cracks and being interesting. So. Alright.
cool. Thank you.
Thank you very much.
Just doing some changeovers here as we're coming up towards the hour. sorry? Adam. Right. Cool. Yeah, these kind of wider views are quite helpful to scan around and look for those dark rock areas, but from what I've seen so far, a lot of the venting seems to be coming out from these kind of ledges and things. Hello. Well, that's the end of my shift, so I'm going to sign over to Jorge Cortez from the University of Costa Rica. Thank you, everybody. Oh, I'm taking the pokey stick.
Hello, hello. Hola. Well, good afternoon, good evening, good morning. It's Jorge Cortez from the University of Costa Rica, and I'll be here for the next hour. We'll continue doing some exploration here. Buenas tardes, buenos días, buenas noches. Les habla Jorge Cortés de la Universidad de Costa Rica y vamos a continuar aquí explorando este sitio llamado Caballito y vemos una gran cantidad de organismos aquí.
For you, Adam, do you have automatic roadless stabilizer, or do you have to do everything manually?
We have a little bit of both. We have auto heading, so it allows us to walk in the heading. We also have what's called station keep, which also lets us more or less hover in one area, but a lot of times we just fly manually.
We're looking for this.
The vast majority is manual.
What's this? Is this cruise control? Oh, that's
Okay, we're seeing here big octocorals, sponges, and some of these octocorals covered with whittle stars. Aquí vemos esponjas de esas de vidrio, octocorales, y los octocorales cubiertos con una gran cantidad de estrellas frágiles, y vemos diferentes tipos de octocorales como estos que son unos látigos largos, a otros que son más como abanicos. Una gran diversidad de este grupo. Y tenemos con nosotros al especialista Dalis Cabridi de la Universidad de Costa Rica, que está estudiando y describiendo especies de estos grupos. I've seen here a lot of many different octocorals, some that are like a whip, a long whip, others more like a fan, and we have with us Dr. Odalis Capridi from the University of Costa Rica, which is world authority of this group in this region of the world. And she's been collecting and will be working on these species. See some shimmering?
You saw shimmer?
You know, we're going around and looking for shimmering water. It's that you see like a distortion because of the different temperature in the water, and that's indication of heat coming out. Let's see. Let's take a look at that. And we're seeing a acorn worm, I think. We're gonna settle down, take a look at this worm. Wow. This is a group of called hemichordates, the same line of evolutionary line as we are, and you can see the digestive tract and how they absorb sediments, get their food, and how it's accumulating at the other end, and eventually will come out with that shape. O sea que lo que vemos es este gusano bellota que llaman, y se le puede ver bien el tracto digestivo donde toman sedimentos que lo van procesando, retirándole el alimento, y a donde se va acumulando atrás antes de botarlo al exterior, y salen con esas formas. And it looked different from the one we've been seeing in the abyssal plains, which is purple and different shape. Good, beautiful. Yeah, that acorn worm looked like an orchid. And we keep moving. And as I mentioned, one of the objective of this expedition in Octopus Odyssey aboard the RV Falcor 2 of the Schmidt Ocean Institute is to look for warm water coming out from these rocks or sediments, and the effect it have on the organisms around. And we found that there are nurseries, octopus nurseries, where this warm water is coming out. So we're looking for that, and the way you notice is because there's a distortion in the water. The water will be shimmering, as we call it, and it's a distortion because of the difference in temperature. Right now, the ambient temperature down there is 1.84 degrees Celsius, and this warm water could reach up to 15 degrees Celsius. So it's doesn't sound that warm, but down there is very warm water. Uno de los objetivos de esta expedición llamada la Odisea de los Pulpos, a bordo del barco Falcor 2, Falcor también, del Schmidt Ocean Institute, y lo que buscamos son estos lugares donde está saliendo agua más caliente, que eso significa que hay actividad ahí abajo, el agua se calienta y sale, y en realidad no es agua tan caliente, pero contrastado con el agua que estamos aquí donde estamos ahora, que está a 1,87 grados centígrados, 1,86 ahora, y esa agua que está saliendo sale entre los 10 y 15 grados centígrados. Entonces hay una diferencia muy grande en la temperatura. Vamos a observar esta estructura, era así, de una antigua esponja y con una esponja de vidrio viva al lado. What?
Is that thing in the middle on the left?
That?
You know what it is?
No, we'll take a look at that. Aquí vemos what we see here is like that sponge with a live one, and we're gonna take a look at something over here. That's what you're looking at, or not
No,
that
I
one?
was looking this, and now I just spotted this as well.
Okay, so let's first take a look at this.
Maybe that one is not going to work.
What you want to look at?
Yeah. It looks like a Okay, yeah, that's an ascidian or a tunicate. You can see one of these siphons where water either goes in or comes out. Uh, this is a group also on the same evolutionary line as we are. Let's take a quick look at this over here. Whoa. Here, what it looks like some sort of worm. Not sure what it is. Lo que veíamos antes era un tunicado, la bola con unos sifones, y ahora lo que vemos es una especie de gusano. Es un gusano cuchara, no estoy seguro, que no habíamos visto antes. Okay. This one I'm not sure. That was the other option I was thinking of, but oh no. It's nice. No, but that's a bryozoan. That's beautiful. Extremely delicate creature. It's almost impossible to collect. If we pick it up, it will just break up. So let's do some good video of that. Most animals. That's nice. And let's. There was something else up that way. That. Oh, that looks like a. Seems to me like a stylaster. calcified hydrozoan.
This
Yeah, I think that would look very much like a stylaster. We'll try to collect later on when we're in the collecting phase. If we see one, we'll try to pick it up. That's another very, very delicate skeletons that break up very easily. Eso que vimos antes, como un coralito, es probablemente un estilaster, es un hidroides calcificados, le dicen hidrocorales. Son especies muy muy lindas, pero también muy delicadas. Eso es muy difícil recolectarlos porque se despedazan muy fácilmente. Ahí vemos un pez en el centro del cuadro, esponjas de vidrio. Vamos a ver, take a look at what is this.
Where'd you see it? Oh, right here.
We want to check out something here that we're not sure what it is. Here we see a squat lobster that seems to be dead. Whoa. Not sure.
looks like worms, maybe.
Yeah. But one worm. There's a group that has these very long tentacles. Wow.
Like a spaghetti worm or something.
Yes, exactly.
Okay.
Very nice. Yeah, there's another one of those. Oh, beautiful. Yeah, it looks like a stylaster. It's calcified hydroid, also called hydrocorals. So aquí vemos bien ese hidroide calcificado, estilaster, muy hermoso. Beautiful. Yeah, it seems relatively abundant here. As you can see, as we zoom out, seems like there's not much there, just the big animals, but once we zoom in, the rocks are completely covered with organisms. Podemos ver cuando nos alejamos y quitamos el zoom. Solo vemos los organismos más grandes y pareciera que no hay mucho en las rocas, pero Cuando nos acercamos y vemos las rocas de cerca, vemos que están tapizadas de animales. Y además de una cantidad grande de microorganismos. Yeah, we're moving up the outcrop. Depth here is 2,994 meters.
Range updated position.
Right.
Copy that.
It's a cool view everywhere. Huh? It's a cool view. About that. Little octopus.
Mm.
And that was 600 feet. Drop this off in January. So how like treatment courses, yeah, electric treatment courses, you can get 50. Yeah, people get electric treatment courses. Depends on the audience. Yeah. Shot that certificate, something to get courses. But normally they get tests, physical tests. Your body can handle pressures, etc. From start surface to 320 feet, 4 months.
You were going into a bell or what? Then you're going into a bell?
Oh, it's a lot.
Well, how are you doing your decompression of this thing?
For anyone just tuning in right now, watching from home or from a classroom or wherever you might be joining us from, you are watching live footage from the remotely operated vehicle SuBastian, currently shy of 3,000 meters below the surface of the ocean. And today, this is dive number 539 for Sebastian, but it's our 11th dive for this expedition, the Octopus Odyssey. Currently cruising around looking for some evidence of venting, which if you've followed along on any of the previous dives of this expedition, we're kind of looking for shimmering water, similar to the shimmer you'd see over a hot piece of asphalt or like the hot hood of a car, that heat shimmer.
Aquí seguimos explorando con el vehículo operado remotamente, el SuBastian. Estoy aquí en la sala de control con pilotos Adam y Ben. Y esa otra persona que lo oyen narrando es uno de los cargados de divulgación aquí, que se llama Alex.
Bridge, I'm going to move you another 30 meters west. It's starting to flatten out over here, so we should be making forward progress.
Yeah, copy that, Ben. That's all good.
The big one right below.
Yeah, we moved into a different environment. There's a lot more sediments here, covering the rocks. And when that happens, we don't have so much microfauna. But if we were to take a sample of those sediments, analyze them, there are thousands of species there of bacteria, small animals, fungi, and probably other groups like archaeas and viruses. So everywhere we look, there's life. And here we have one of the big characters in this expedition, and it's been this tripod fish. I've never seen it before in other expeditions to the deep water of Costa Rica. This is my first time, and it's a really spectacular fish. You could see it standing up on its fins. They're modified to walk on the bottom, and it really walks. And it's called a tripod fish. You can see also the pectoral fins have a very long ray. Ese es el pez trípode, que ha sido uno de los personajes importantes que nos hemos topado en esta expedición. En todos los expediciones anteriores, aguas profundas, nunca había visto este pez llamado pez trípode por razones obvias, y lo que vemos son las aletas modificadas con esas extensiones que parecen que fueran pies, y camina sobre el fondo, pero también tiene la capacidad de natación. Aquí podemos ver el detalle de las aletas pectorales, las aletas de adelante, que tienen unas extensiones, unos rayos muy largos. Ahora se ve mucho mejor. Yeah, here we can see the details of the pectoral fins. It is impressive view.
Tell me all the way in. That would be great. Yeah, if you just hold it up a little bit for 5 seconds, 10 seconds, and then let them in soon, and then all the way up. Yeah, perfect.
Tell me.
That should be it.
Like that. Beautiful. Beautiful views.
That's good. Sweet. I get it. And Octopus Odyssey does have a hashtag, so if you're tweeting screenshots or just talking about the dive, or maybe you want updates, you can't be watching the stream all day maybe, you can follow hashtag Octo Odyssey. That is, there are 2 O's in the middle of that. It's not Octodyssey, Octo Odyssey. On Twitter, we'll get you a little feed about this cruise.
Y si quieren darle seguimiento a toda la expedición en el sitio Octo Odyssey, OCTO-ODYSSEY, Octo Odyssey. Ahí pueden ver tomas anteriores o lo que estamos viendo en este momento. Let's see how thick is the sediments in here.
For me?
Let's see how thick is the sediment. So sit down and with Excalibur see. Let's give it a try. So it probably might be just a little layer.
Go for an Excalibur.
Yeah, we're gonna settle here for a minute to poke sediment, see how thick the layer might be.
I don't know if you want to turn around. You're so far away.
Oh, no, not with the probe. We just want to see what other centers.
Wow, that's all right there. It's
That's it.
How deep is that on there?
Probably 7 plus centimeters. Let's see.
Whatever piece of the tape.
Yeah, we'll count when we come over. About 60 centimeters, yeah.
It's like 40 centimeters.
Yeah, that's pretty tough. Like especially because by the probe, it looks like there's some rocks or
Yeah, let's try another.
yeah, maybe try another spot.
It looks more around that area.
Can we come off bottom and just pan around?
If you haven't seen the use of this stick before, this is what the pilots and the scientists will refer to as Excalibur from time to time. This is used for sort of figuring out both how deep the sediment is, but also how soft the sediment is. So as they press it down into there, each piece of tape is 10 centimeters apart, or notates 10 centimeters. And we are kind of just pressing that down and seeing if we can get it all the way down there, or if it starts to push the ROV up, us trying to push that down. We've been using this a lot for either determining if an area is appropriate for core samples, sediment samples, or also for the heat flow probe. Because the heat flow probe is actually quite delicate itself. So we don't want to try and jam that down into sediment that's a little too hard or too rocky. So we can test with Excalibur, which I think it's just a rod of metal.
Well, we pulled it from a stone.
Yeah, Adam's telling us that it was pulled from a stone, of course, but.
Sí, eso que vieron antes es una varilla de metal marcada cada 10 centímetros y se usa para medir la profundidad del sedimento, y si la profundidad es suficiente, entonces podemos hacer mediciones de flujo de calor. Si no, no bajamos el otro instrumento, que es muy delicado y se puede dañar. El instrumento es para medir la profundidad, lo llaman Excalibur. Era la espada del rey Arturo, y aquí están haciendo bromas que eso fue sacado de una roca, como en la leyenda. Pues es un pez diferente.
Big difference from what we used to down here.
Yeah, that was a different species of fish, which I haven't seen before, especially down here. Vamos acercándonos a esta langosta. Ujú. Muy bien. El grupo de los crustáceos. Yeah, that was a good view of a squat lobster, and we saw a stylastro on the back, on the rock, and here we have sea urchin on the left and sea urchin on the right. Oops, me, a sea cucumber on the left, sea urchin on the right. Ahí veamos un pepino de mar, ese morado ahí, que es un equinodermo. Aquí vemos una estrella sobre algún otro animal que es otro equinodermo, y más atrás a la derecha había un erizo de mar que es otro equinodermo. Es un filo muy abundante en las aguas profundas. Yeah, we just saw 3 different classes of echinoderms. It's a very abundant phyla in the deep waters.
Ready to updated position.
Drop it back.
Might be able to get a probe up ahead here.
Let's try it. Let's try it. Yeah, it looks.
What I'll try is I'll go to waypoint 9 there. It looks like it flattens out there.
Uh-huh.
As we fly over this sediment here, it might look like there's not a lot here, but anyone who's been around while we've done the heat probe knows that as we zoom in, while we're sitting there doing the heat flow probing, there's quite a lot moving around on and in that sediment.
Como acaba de explicar Alex, pareciera que no hay mucho en estos sedimentos, pero cuando se enfoca de cerca vemos una gran cantidad de organismos. Aquí estamos viendo uno de estos gusanos bellota, como hemos visto en otros lados, el pez trípode, que ya se ha vuelto relativamente común de verlo aquí, y al extremo derecho un pepino de mar. Ahí lo vimos nadando. Y aquí vemos el gusano bellota, y de nuevo con su intestino transparente, entonces podemos ver a donde está moviendo la arena y ahí a donde ha salido ya unos pocos. Muchas gracias a todas las personas que están conectando, enviando saludos. Saludamos de vuelta. Thank you everyone sending their message, telling us where they're writing from. So we're gonna do another probe of the sediments here.
This would be Adam.
Okay. Fairly steep. Nope, no. Still up, it's just I don't know if I feel it too much. I need water there.
Okay. Yeah, the sediment on the bottom is very, very fine, and so it's just landing steers. It's mud storm. Yep. Los sedimentos del fondo son muy, muy finos, entonces con solo acercarse, acomodarse para hacer una medición, hace que se levante una nube de polvo. That's it. No. Yeah, sediments are not thick enough, but that was like 30 centimeters. So we'll just move on.
009 or passing waypoint.
Yeah. Let's move and we see another sediment patch. We'll give it a try again.
I'll just jump up here like maybe 50 meters or so. It seems
Yeah.
Like there's a crust underneath it, I think.
Mm-hmm.
You might have just heard that we're passing waypoint 9. So I've pulled up the little map here. And you can see waypoint 9 right there in the middle. And we are headed to the left from there. So we came up that nice little slope and we are going to kind of keep heading up. Perhaps a little more gradual than what we had earlier. But I'm sure we'll see some variance here and there.
Alex, let me do that same, like I'll say in Spanish.
Yeah.
That look really neat. Aquí estamos viendo el afloramiento y ese es un acercamiento de la ruta. Esos son los puntos que se marcan, lo que llamamos waypoint. Y ya estamos pasando el 9 y veníamos en esa planicie y subimos esa, ahí se ve muy bien en esta perspectiva, subimos esa pendiente que vimos antes, las esponjas y otros organismos, y ahora estamos moviéndonos hacia la cima con una pendiente más leve, y apenas las pendientes más leves vemos capas de sedimento más grueso. Vamos a ver si más adelante encontramos unas capas de sedimento más espesas para hacer las mediciones de calor. That's good. Thank you. Sé lo que esa imagen que vemos, esos son se construyen de batimetría que hace el barco en la noche, que se mueve de un lado a otro con una sonda y de ahí sale, se hace esa construcción de ese montículo, ese monte submarino, y lo podemos ver desde diferentes ángulos, y sobre eso se establece la ruta para seguir durante el día.
Yes. How's busy? I got autos on there's a little bit cruise already.
Yeah, we saw was that image of this outcrop, and that's generated by the bathymetry that was done last night, and the ship just moved up and down with a multi-beam scanner and scans the bottom, and all that information comes back, it's computed, and we reconstruct that structure of the outcrop, and we can see it from different angles. And on top of that, we put the transect that we want to follow, and those are the waypoints. And now we just passed waypoint 9, moving towards waypoint 10, going up the hill. Right now at 2,937 meters. Entonces seguimos subiendo poco a poco, aquí donde hay acumulaciones de sedimento, y aquí estamos a 2.937 metros de profundidad. Okay, my time here is over. Thank you for being with us. This is Jorge Cortez from University of Costa Rica. Thank you for joining us, and I'll give this science seat to Celeste Sánchez, also from the University of Costa Rica. Bueno, muchas gracias por habernos acompañado durante esta última hora. Es Jorge Cortés de la Universidad de Costa Rica. Me retiro ahora y le cedo el ciento de científica aquí que siga guiando la misión a la doctora Celeste Sánchez de la Universidad de Costa Rica. Buenas tardes.
While the scientists are doing their handover there, we've also got 2 new pilots in. We've got Chill piloting Sebastian right now, and Mike next to me, currently working the manipulator. And then, of course, my name's Alex. I'm the multimedia technician on board, and for much of the day shift, I am in the data logger seat, just taking notes of the incredible sites we're seeing, the sites we're able to share with you, and sites we would love to keep sharing. So, all those nice little zooms you see, and you might hear me whispering at the pilot like, zoom in more, turn off the lasers, right? That's what I do. But at other times I have to get up, and we do have programs that the ship offers to classes and groups around the world. And I think that's one of the things that makes our work here very special, is this is deep sea science that we can share live while it's happening. Either out to living rooms or classrooms or lecture halls. Anywhere in the world you got internet, you can join us and explore the deep sea live. And we appreciate everyone who does. And if you have any questions while you're watching, feel free to type those in the chat, and we do try to answer those as we can.
Hi everybody, my name is Celeste Sánchez Noguera. I will be with you for the next hour. I'm from the University of Costa Rica. Hola, buenas tardes. Muchísimas gracias por estar acá con nosotros. Mi nombre es Celeste Sánchez Noguera de la Universidad de Costa Rica y voy a estar con ustedes durante la próxima hora de este buceo. Estamos en este momento en el monte submarino conocido como Caballito y en este preciso instante estamos justo en uno de los fondos de sedimentos haciendo pruebas de profundidad con el Excalibur para ver si tenemos suficiente profundidad de sedimento para hacer más mediciones de heat flow. Pareciera que sí, y en ese caso procederíamos a hacer las mediciones. El objetivo, o uno de los objetivos del buceo del día de hoy, son precisamente tratar de hacer una caracterización del flujo térmico que hay en esta zona, y por eso es que tenemos que realizar estas mediciones de heat flow. Además de eso, seguimos buscando algunas de las especies objetivos que todavía nos faltan recolectar. Y este es un sitio que no se había explorado previamente, así que toda la información igual que se está generando es información de muchísimo valor. So I think we're going for a heat flow, right? Okay. So it seems like it's a good spot for a heat flow measurement.
All right, moving down.
And usually it takes about 15 minutes for each measurement, which means that we will be here in this specific spot for about 15 minutes, and then we will use the time to just look around in the sediment for all the fauna that is usually living in association of these soft bottoms. Vamos a comenzar en estos momentos la medición de heat flow. Eso toma aproximadamente unos 15 minutos. Así que aprovechamos este tiempo para observar con las cámaras del submarino toda la fauna que vive asociada en estos fondos de sedimento. En estos momentos estamos a 2.937 metros de profundidad con una temperatura de 1.83 grados Celsius. Y en el fondo vemos de hecho bastantes señales de que han pasado pepinos de mar por acá. Por medio de las excretas que vemos. What is that hole over there in front? It's something that the submarine did, or
Sorry, what?
there was like a hole in the bottom, like
Our side. There's a ridge on the bottom. But I mean, there's kind of a slide on the slope. Someone? 6.6.
Les recordamos que pueden seguir estos buceos por los diferentes canales de YouTube del Schmidt Ocean Institute y que todos quedan grabados. Así que si en algún momento quieren ir y revisar alguno de los buceos de los sitios anteriores que hemos estado explorando durante esta expedición, también lo pueden hacer sin ningún problema. Además de eso, hay blogs escritos por varios de los participantes de la expedición que están disponibles tanto en idioma inglés como español en el sitio web. You see that hole over there? Is that something that the submarine did? Or no?
Oh, yeah, we've seen like little disturbances like this.
Yeah.
Yeah, that's where I landed right there. And
Yeah.
I just pivoted around, so I don't think that was for me.
Yeah. I mean, there's been times where we're doing like transects across areas we haven't been, and we're finding these little dugout depressions.
Okay.
We have yet to catch what's doing it.
Yeah, exactly, because it's really, I mean, it's a very irregular shape, and
Yeah. Sometimes it's us. You know, if we've been spinning around in a circle, that can be us. But we've seen them where we're just going in a straight line and it's there.
Okay.
It does look like something I did, but
Yeah.
I don't think that's where it was.
Yeah.
'Cause look, there's no disturbance around it.
Yeah.
I don't know.
Okay.
Not sure. I'm already in here, so.
Jury's out
Estamos
on whether that one's us or not.
preguntándonos si esto, este hoyo que acabamos de ver en el sedimento fue hecho por el submarino o si fue hecho por algún animal. Y los pilotos nos comentan que de hecho en otras inmersiones han encontrado cosas similares mucho antes de que el submarino toque fondo. Entonces podría ser algún animal, pero todavía no hemos logrado tener la suerte de determinar qué es lo que lo ha podido hacer.
So this
Okay,
is
like a brittle star that was just
yeah,
Yeah, I was just thinking when you take your kids to the beach and they like cover themselves.
Yeah, exactly, something like that. Yeah, exactly. It looks like it's kind of struggling actually with that sun, but yeah. Okay, let's see what else do we have here. What is that? Is that a sponge? No. It's just like a piece of clay. Hard to focus, yeah.
I don't know. It might be a sponge.
Yeah.
Or there a piece of clay.
Yeah, I think it's a piece of clay.
Yeah.
Yeah, it is a piece of clay. Well, there is one shrimp over there. Tiny shrimp. En muchas ocasiones, durante este tiempo de espera, mientras se hacen las mediciones de heat flow, hemos logrado observar organismos bastante interesantes. En otras ocasiones no hay tanta fauna, pero sí vemos cosas como, por ejemplo, estos hoyos misteriosos. Esto que están viendo en este momento son excretas de pepino de mar. Así que para nosotros esto es un indicativo de si no estuvo el pepino ahí, que efectivamente hubo pepinos en esta zona recientemente. Los pepinos de mar lo que hacen es que se alimentan de muchas de las bacterias y los microorganismos que viven en la arena. Entonces ellos toman las partículas de arena, las ingieren y una vez que ya han absorbido todo lo que es alimenticio para ellos, you see, it's like there is something still there, right?
I think it was falling. It was collapsing.
Yeah. Okay.
And our thrusters are just full of
Okay.
water, I
Okay.
think.
Okay. Entonces, muchísima de la arena que se encuentra en realidad en el fondo marino y también en nuestras playas en algún momento ha pasado por el tracto digestivo de un pepino. Can we have a look at that octocoral, or is too far away? Yeah, okay. You can just focus on the star and see how long does it take to actually get rid of the sand.
Maybe it doesn't want to get rid of the sand.
Is it digging into the sand?
No, usually they don't dig into the sand.
Because I've seen several of them in holes, and I'm assuming they're
Yeah?
digging it or just.
They do bury
Following.
themselves beneath the sediment,
Yeah.
and then they pinpoint that way.
I like the ones that are like buried, but you can still see the outline.
Yeah,
Yeah. Yeah.
It's like, you're not doing a great job.
Yeah.
Yeah.
It's like it's doing a dance routine.
Yeah.
I think of it like a snow angel.
Snow angels. Yeah. This one's the marine snow angel.
Yeah.
Yeah. The ooze angel.
The ooze angel.
You know, like the dogs that they'll hide from you, but they only hide in their eyes. So they
Yeah,
think
the rest
they see
is,
you.
yeah.
Their whole body's hanging out.
Yeah.
Yeah.
That's my dog. She's not very smart.
Yeah.
Hicimos una exploración rápida y parece ser que no hay otros organismos ni grandes ni pequeños que observar, así que decidimos quedarnos enfocando a esta estrella quebradiza que está enterrándose en el sedimento. Y bueno, como pudieron escuchar, un par de comentarios vacilones al respecto que se han generado acá. Algo que es bastante importante de este tipo de expediciones es que realmente nos brindan la oportunidad de poder explorar en detalle estos ecosistemas de océano profundo a los cuales usualmente no tenemos acceso fácilmente sobre todo por el hecho de que en muchos de nuestros países no tenemos barcos de investigación oceanográfica con la capacidad de poder realizar este tipo de expediciones. Así que en ese sentido la colaboración científica que se puede hacer con colegas de otros países es realmente muy importante. Y en esta expedición particularmente tenemos de hecho 8 investigadores de Costa Rica, además de un investigador de Portugal y una investigadora de Trinidad y Tobago. Entonces es un equipo no solamente interdisciplinario, sino también bastante diverso en términos de las nacionalidades que lo conforman.
So you've mentioned that like part of this probe is waiting for frictional heat to go away, and then so
First 7 minutes.
to what degree, like how many points are we measuring on these things?
We're measuring on five thermistors.
Yeah.
Once per second.
And we're measuring once per second throughout the 15 minutes.
Yeah.
Yeah. And I mean, the over-the-side probes that were used previously are 3 meters long, and the new ones are 5 meters long. So, you need more thermistors.
Yeah. What's the resolution of a degree that this thing can see?
It's more the difference that matters. If you're measuring a gradient, and it's less than a tenth.
Okay.
It was also made in the 90s. So.
I like that caveat. But just so we're clear.
But the gradient, it doesn't care.
I enjoy that. All right. If you missed that, apparently it was an item of note that the heat flow probe was actually made in the 90s. Still does its job, but just a note.
I bet a lot of the scientists on board were made in the 90s too, and they seem fine.
That's it. That's it.
Excellent
Yeah.
observation.
Or the 80s.
Yeah.
Or before that.
Yeah.
Un dato curioso sobre el sensor que se está usando en este momento para medir el flujo térmico es que fue construido en los 90s, así que se han hecho comentarios aquí de que es interesante ver que un sensor tan viejo relativamente todavía pueda estar funcionando tan bien, pero igual eso sucede muchas veces con la ciencia. Si se construyen instrumentos de la forma adecuada en realidad y se les da el mantenimiento correcto, pueden durar por muchísimos años funcionando adecuadamente.
I am curious now, though, what the oldest piece of equipment has been on SuBastian, as far as gear. Yeah.
I mean, most of the tech that gets developed for seafloor operations, you want to make it as Luddite as possible, because that simplifies the number of things that can break. And for example, a lot of the corks, seafloor observatories are connected through RS-232 connections, like 9-pin. So you have to use an old computer or some kind of a dongle to downgrade the connection.
Yeah.
Y efectivamente, pues muchísimo del equipo que se hace para trabajar sobre todo en aguas profundas, generalmente se construye pensando en que sea equipo bastante duradero y que ojalá no requiera un mantenimiento muy complejo. Sí es importante sobre todo que sea equipo muy resistente porque va a estar funcionando en condiciones bastante extremas de presión y de temperatura también. De hecho, uno de los instrumentos que hemos estado colocando en diferentes lugares, que se llama Osmosampler, tiene un funcionamiento muy interesante porque lo que hace es capturar o recolectar muestras de agua de forma continua por medio de un proceso de difusión. Entonces, básicamente lo que usa es un principio de difusión por medio de un gradiente de concentración de sales y esto se conecta a un tubo de teflón de un diámetro bastante delgado que puede llegar a medir hasta 200, 300 metros y eso se deja en el fondo. Entonces es un sistema que funciona sin baterías, sin ningún tipo, digamos, de energía externa, porque de hecho los instrumentos con batería realmente no funcionan a estas profundidades. Y es un sistema relativamente sencillo, pero que ha resultado muy efectivo para poder recolectar muestras de agua de manera constante en este tipo de ecosistemas.
Sorry guys, any idea how much longer? Using a little bit more thrust than normal, and I'm starting to get warm.
We're good.
We're good. Okay. Open up.
Okay, so we just finished with the heat flow measurement, and now we are going to start to move and continue our way to the next point. Acabamos de terminar en este momento la medición, así que vamos a comenzar a movernos para seguir desplazándonos hacia el siguiente punto de muestreo. Estamos en este momento justo a la mitad entre el punto 9 y el punto 10 del plan de buceo del día de hoy, que incluye un total de 16 puntos. Así que estamos un poquito más a la mitad. Les recordamos que el buceo de hoy está planificado para terminar alrededor de las 4 de la tarde, hora local, lo que significa que todavía nos quedan aproximadamente 2 horas y media más de buceo.
Opportunistic. If there's sediment, great. If not, we're just keeping transect.
So we just keep moving to the next waypoint, right? Okay. What was the thing about the temperature?
I was using a lot of power to stay down, and so the motor gets hot.
Okay. Aparece ahorita en el transecto una estrella de mar de color rosado, nos recuerda un poco a Patricio de Bob Esponja. Tenemos un pepino, de hecho este pepino fue una de las especies objetivo que se colectaron o que se pretendían recolectar. I think we're ready for that one, right? Yeah, that's not the one that we are aiming for. Okay. You want to take that frame back? You got it? Okay. Yeah. Thank you. Los pepinos de mar realmente han sido parte de las estrellas de esta expedición, sobre todo en estos fondos de sedimentos. Hemos podido observar una gran diversidad de pepinos y algunos de estos se han identificado únicamente con fotografías, pero otros sí los hemos recolectado para poder hacer una identificación más apropiada en el laboratorio. Recuerden que muchísima de la fauna que se encuentra en ecosistemas a estas profundidades no ha sido previamente descrita, así que en muchos casos es necesario poder recolectar los individuos para poder hacer la identificación de regreso en el laboratorio. En el caso de esta expedición, se cuentan con todos los permisos de colecta otorgados por las autoridades nacionales para poder recolectar un número determinado de especímenes, y todos estos organismos serán depositados posteriormente en el Museo de Zoología de la Universidad de Costa Rica, que es el repositorio oficial nacional para todas las muestras científicas. Esto lo que permite es que pues tanto los científicos de esta expedición como cualquier otra persona que en un futuro quiera poder consultar las muestras puede hacerlo sin ningún problema, porque son muestras que quedan en realidad a disposición de la comunidad científica. Y pueden ver ahí más evidencia de excretas de pepinos, que era lo que les comentaba hace un rato. En realidad son organismos bastante comunes de encontrar en este tipo de fondos de sedimentos. I think they still want a crinoid, right? Yeah, so can we try to get it? Do we have... Okay, we haven't used any of the coral quivers. Just check. Right. Esto que tenemos en la imagen es un crinoideo. Just double checking with Beth if we still want to get the crinoid. Maybe you can just take like frame wraps. Did you get the close up, or is it still too blurry with the sediment? No, it's okay. Okay, so you just need with the lasers, right? Yeah. Okay, so if we can take one with the lasers on, please. No, we are on transit to waypoint 10. That's a really nice close-up. Yeah. Yeah, that's really nice. Okay, no. She says that we should be moving and then just get to the next waypoint.
Yes.
Yeah.
Jess is moving all the time. We're moving.
Yeah.
Perfect. Got it, Alex? Yeah.
Sorry.
Yeah.
No, that's all right.
So Ben was a sea star. Breathing.
A breathing what? Breathing Jitsi start. And what is that? Okay, just let me check. Al parecer eso no era un crinoideo, sí es un equinodermo, que es de la misma familia de los pepinos y los erizos de mar, pero no era un crinoideo, sino un tipo de estrella. Yeah, that was our mistake. That was not a crinoid. That was a recently start. They are still of the same group of the echinoderms. So we are just very close to the waypoint 10, and once we get there, we will again see if it is possible to conduct some heat flow measurements there, and if not, then we will just continue moving in our way to the next waypoint. Estamos en este momento llegando al punto número 10 del transecto del día de hoy. Una vez que lleguemos, vamos a probar si es un buen lugar para hacer mediciones de flujo térmico, y en el caso en el que no se pueda, entonces vamos a continuar la travesía hacia el siguiente punto. No, yeah, exactly. Yeah. So vemos en este momento en pantalla el Excalibur, así que vamos a probar si tenemos suficiente espesor de sedimento para poder realizar la medición de heat flow. Recuerden que esta varilla tiene 1 metro de largo, así que aquí sabemos que con seguridad tenemos al menos 1 metro de profundidad, lo cual es el mínimo requerido para poder realizar la medición de flujo térmico. So, it seems good, right? Yeah.
Oh, this is way from 10.
Yeah.
I thought it was.
Yeah.
But we only got 40 meters from the last station. So, sorry, you can. Yeah.
Okay, so do you want to continue?
That was my miscommunication.
We were like halfway between this.
Yeah.
and
We
this.
just
hit everything off.
like every 100.
Yeah.
If there's a
Okay.
place we're happy. If not, we're also happy.
Okay, so do you want us to stop like midway between the next two?
Yeah, because we want it at least 100 meters between measurements.
So the confusion here is that we have been measuring at waypoints, but we got to waypoint 9. I don't think there was like ideal sediment there. So we continued on the track, and we ended up like halfway between waypoint 9 and 10, did the heat flow there. And so now we're going to wait again until we're a little ways past waypoint 10, which we're at now, to do another heat flow, because it's just too close to where we did our last heat flow.
Okay, at least, but then that means that if we continue doing it like halfway each waypoint, at some point we will reach just toward the end something less.
That's
So,
fine.
yeah? Okay.
Yeah, just it's if they're too close, it's like taking the same.
Yeah, okay.
You won't see it change.
Okay, perfect. Vamos a continuar desplazándonos. Aquí sí teníamos la profundidad requerida para hacer la medición de flujo térmico. Sin embargo, necesitamos que los puntos de medición tengan una distancia mínima de 100 metros entre ellos. Y como la otra medición la realizamos justo a medio camino entre el punto 9 y el punto 10, entonces vamos a desplazarnos a nuevamente la mitad de camino entre lo que sería ahora el punto 10 y el punto 11 para ver si podemos realizar ahí la medición. possible to use like sonars or something like that to actually know the exact thickness of this sail? Okay, but there are some other methods that can be used, right? And usually they are just like something. Sure.
It's basically
Yeah, something. Yeah, yeah. Okay, but it's like more or less sonar signal, something like that. Okay.
It's just lower frequencies.
Yeah, a different. Okay. Okay, thank you. En algunos barcos, no es el caso de este barco, pero algunos barcos están equipados con equipos que son un tipo de sonar que pueden emitir ondas a diferente frecuencia, y esa es una de las maneras en las que se puede estimar a veces cuánto es la profundidad o el espesor del sedimento en el fondo marino. Pero en este caso, como les mencionaba, no es un equipo con el que contemos en este barco y no es algo, digamos, que necesitemos para los objetivos de esta expedición. Oh, podemos ver en este momento, además de los pepinos, más peces trípode. Estos han salido o han sido bastante frecuentes en este buceo del día de hoy, en esta zona abisal de fondos de sedimentos. Y realmente son especies bastante interesantes. Parecen, como decíamos en algún momento, sacadas como de una pintura de Salvador Dalí. El nombre común es pez trípode, precisamente porque tienen sus aletas pectorales y la aleta también anal modificada para que sean más alargados y se ponen en esta posición sobre el sedimento. Este pez puede probablemente pertenecer al género Batipterois. So the reason why we are not doing the heat flow measurement on specific on waypoint 10 and we are just moving to half of transect is because they need to have a distance between measurements of at least 100 meters. Otherwise, the measuring points are too close and it's pretty much like measuring exactly in the same point. So it had something to do basically with being able to know that we are actually measuring two different points and not something that could be giving us the signal of the same point. So should we give it a try here?
Yeah, we're real close.
Yeah.
I'm just letting the boat get a little closer so I don't have to
Okay.
switch this way. Oh, is it just? Oh, never mind.
No, I
That's the
think
negative.
this is the reflection of the... Yeah, okay. Estamos llegando en estos momentos a la mitad de la distancia entre este punto y el próximo, así que vamos a tratar de ver si aquí sí podemos realizar una medición de heat flow. So we are just now halfway from the next waypoint, so we will try to see if it's a good spot for a heat flow measurement. I'm just trying not to stab that sea cucumber with the Excalibur. Y perfecto. Tenemos al menos 1 metro de espesor en el sedimento, lo cual significa que sí podemos hacer una medición de heat flow y que vamos a estar acá al menos 15 minutos durante los cuales vamos a aprovechar para tratar de explorar con las cámaras los alrededores del submarino.
I'm stable. Okay, recording. All right, probing down. Probe is down.
Ok. El sensor de heat flow ya se encuentra en el sedimento.
The only thing I could say is I did take a little extra time, so I'm not pulling from the vessel. Let it stabilize. I was using too much thrust at the last site, so maybe that's what it is. It took me an extra minute or two, but
Yeah, it makes it look different right there.
I'll keep trying to do that.
De hecho, pueden ver en la esquina superior izquierda de sus pantallas, eso que se ve es la cámara que indica dónde está en este momento el sensor que estamos utilizando para realizar la medición de heat flow. Okay, let's see if we have something nice. Besides from the sea cucumber, of course.
Oh, okay, so don't look at the cucumber. All right.
They're such sweet little things.
I mean, they're really nice, but
They're just doing their life.
they're just
They're the little Roombas of the sea.
like, you know, like poop machines. That's all what they do,
Yeah.
right?
Hey,
Like,
hey, to be fair, they have the most complex digestive system.
I
of all
know,
the Islander.
I know. I mean, I think it's just like marvelous.
Nice recovery.
Now we're that excited.
I get it. They're cool. I like them.
No, I think also the problem here is like is too much resuspension of sediment, so it's hard to look further than what 2 meters or something like that.
Yeah.
I was even like, man, I'll just creep up so we can have something cool to look at. Just stop looking at it.
Stop looking at them. So these sea cucumbers, this particular type that we've got in front of us, some folks will call them gummy squirrels, because that kind of purple gumminess, but also they've got like a tail on them, like a squirrel would, just popped up. But they are fascinating little critters. They cruise around on the seafloor and suck up sediment, digest everything digestible out of it. And then, of course, they leave behind those nice little coils of excrement that we've been seeing as we've been flying along. I like to think of them as little Roombas of the deep sea. They play a very important environmental role as a decomposer and sort of picking up everyone else's mess.
I have no idea what is that. You want to take a frame round of that? I don't know what it is. It's a crustacean, but what exactly? I have no idea. Muchos de los organismos que viven en fondos de sedimento son organismos que, como les mencionaba antes, consumen muchísimas de las partículas del sedimento, así que son organismos que ayudan también a procesar mucho de lo que son restos de excretas de otros organismos también. Así que son una especie de organismos detritívoros también y cumplen una función bastante importante en términos ecológicos.
I'll name him Speedy.
Speedy, yeah.
I was thinking, this is quite the leisurely stroll we've got going.
Can we get a better close-up, or that's the best zoom? That's okay.
He's just that small.
Yeah.
Need some lasers on it, see what he looks like.
Oh yeah, look at that.
So less than a centimeter, it looks like. Yeah.
Frame wrap with the scale. Yeah. Sí, con la escala que estamos utilizando en este momento podemos ver que este animal puede medir aproximadamente 1 centímetro o inclusive menos. Es realmente pequeño. And there is even a tinier one. Look at that. How long do we have left from the hip drop? How long do we have left from the hip drop?
Still have like 600 seconds.
Okay. 10 minutes? Okay.
Yeah, as long as you're understand that.
Continuamos aprovechando nuestros 15 minutos de mediciones de heat flow para explorar un poco el fondo. Nuevamente vemos bastantes estrellas quebradizas, el erizo de mar el pepino de mar que vimos hace poco. Esto que estamos viendo en la imagen al lado de la estrella pareciera una esponja bastante pequeña. Y esta estrella que ven, de hecho, uno de los brazos está parcialmente cortado. Y en realidad los equinodermos son organismos muy interesantes porque tienen una capacidad de regeneración bastante interesante. I think it will be a sponge as well. I mean, this for sure is a sponge. That one? I mean, if we weren't here at this depth, I will easily say that's one of these algae, like the green algae. Generalmente este tiempo de espera de las mediciones de heat flow nos permite observar con más detenimiento este fondo de sedimentos y poder identificar algunos organismos bastante pequeños que de otra manera probablemente pasarían desapercibidos. Y en realidad hay toda una gran diversidad de animales que también viven enterrados dentro del sedimento. Entonces, una de las cosas que se están haciendo en esta expedición es recolectar núcleos de sedimento para diferentes análisis, y uno de los análisis que se va a hacer precisamente es identificar toda esta fauna que está viviendo enterrada dentro del sedimento. Eso se va a realizar una vez que estemos de regreso en tierra, y van a ser estudios o análisis que van a realizar los colegas de la Universidad de Costa Rica. What is that? Sometimes I wish we can just like poke on things, like. Yeah, yeah. Igual hay muchos organismos que a veces si no se ven completos es difícil saber qué son. Esto que estamos viendo ahorita en pantalla, por ejemplo, podría ser un fragmento de un equinodermo, o también podría ser no sé, tal vez un molusco, pero es difícil decir realmente si no tenemos forma de ver todo el resto del cuerpo del animal. No, but you took a frame wrap? Because I have no idea what it is. I mean, I think it could be like part of a sea cucumber maybe, or a mollusk. I don't know. I think that's too far away, right? Yeah. Yeah. What about there or there? Estamos tratando de enfocar un poco más lejos. Esto que estamos viendo en este momento en pantalla son tubos de poliqueto, y creo que el que está atrás es un octocoral. So this one, it seems to be like a polychaete tube, and the one behind an octocoral. But actually there's something else coming out of this polychaete.
I wonder if that's that weird little spaghetti worm that we saw earlier. It was all stringy.
Maybe. Yeah. muchas, en realidad, de estas expediciones nos permiten precisamente poder descubrir e identificar nuevos grupos, especies que no había manera previamente de saber que existían en estos ecosistemas. En el pasado y en muchas ocasiones las muestras que llegaban, digamos, a manos de los científicos de estos fondos marinos eran producto de dragados o de arrastres que se realizaban, así que eran usualmente muestreos no selectivos. Sin embargo, el poder trabajar con un submarino nos permite ser demasiado, bastante selectivos. Primero, en términos de lo que podemos capturar por medio de videos o fotografías, y eso a la vez nos permite poder tratar de elegir bien qué es lo que realmente vamos a querer recolectar. So at least this part is mostly dominated by brittle stars and some sea cucumbers. Okay. So we just finished the heat flow measurement. So we are just going to keep moving to the next waypoint. Acabamos de terminar la medición de flujo térmico, así que vamos a continuar desplazándonos hacia el punto número 11. Para los que se acaban de conectar, les recuerdo que nos encontramos hoy explorando el monte submarino conocido como Caballito. Este es uno de los montes que no se habían explorado previamente, así que dependiendo de los hallazgos del día de hoy, vamos a decidir si hacemos un segundo buceo en este sitio o si continuamos explorando otros sitios. Hasta el momento en la expedición hemos podido visitar diferentes montes submarinos, algunos donde hemos podido localizar emanaciones de agua, agua digamos levemente caliente, y en la mayoría de los casos en estos sitios hemos además podido encontrar pulpos o comunidades de pulpos viviendo en estos sitios. En este momento lo que estamos haciendo precisamente con estas mediciones de heat flow es tratando de determinar si la temperatura está más fría o más caliente en la corteza del fondo marino, porque eso nos permite entonces determinar si estos montes están funcionando como sitios de recarga o de descarga de agua. En algún momento en los buceos previos les comentábamos que en el fondo submarino existe un sistema de transporte de temperatura por medio del agua. El agua es absorbida desde algún punto, usualmente, generalmente una abertura o un monte submarino que básicamente lo que hace es absorber agua fría, y esta agua entonces es transportada por la corteza o por el fondo, digamos, de la corteza oceánica. Se puede desplazar por varios kilómetros, y entonces en este trayecto básicamente hay un intercambio de temperatura, y esta agua que ingresa con una temperatura relativamente fría comienza a aumentar su temperatura y luego es expulsada por otros puntos, los que se conocen como sitios de descarga, y eso es lo que hemos podido observar, por ejemplo, en el afloramiento rocoso El Dorado. La primera, digamos, exploración que se hizo en estos sitios fue hace 10 años, cuando se llegó al monte submarino o al afloramiento El Dorado, y precisamente eso fue lo que motivó a que se planificara otra expedición, que es en la que nos encontramos en este momento, para poder tratar de relacionar cuál es la asociación que existe o cuál es el motivo por el cual los pulpos se encuentran específicamente en estos sitios donde hay salida de agua más caliente. Creemos que podría ser porque están aprovechando el agua que viene con una temperatura un poco mayor. En mediciones que hemos estado haciendo en los diferentes buceos, hemos podido determinar incluso temperaturas máximas de unos 12 grados Celsius, lo cual son en realidad casi 10 grados de diferencia con respecto a la temperatura del agua en el resto del fondo en esta zona. Así que esta temperatura un poco más caliente podría ser de beneficio para el proceso de maduración de los huevos de pulpo, pero también otra de las hipótesis que se maneja es que la emanación de agua en realidad produce una limpieza mecánica de las rocas. Lo que hace es básicamente remover el sedimento de estas rocas, y que entonces eso provee un sustrato bastante limpio que entonces aprovechan los pulpos para poder colocar o adherir los huevos. For those of you who are just tuning in, we are right now diving in the seamount known as Caballito. This is the first dive that we actually do at this specific point during this expedition, and we are conducting heat flow measurements along a transect of 16 different points. All the waypoints are distance by 100 meters. What is that?
There's a feature coming up here.
Yeah, but that's... Did we know about that feature, Rachel?
No.
It wasn't on the map, right?
Not really, no.
Okay.
It's not that high resolution.
Yeah.
I mean, this happened to us yesterday too, right?
Yeah.
We found it everywhere.
Yeah, exactly.
Yeah, no, this doesn't really show up on the bathymetry.
Generalmente, para elegir los sitios de trabajo, previamente lo que hacemos durante las horas en las que el submarino se encuentra en cubierta es que realizamos un mapeo de la batimetría del fondo submarino, y eso también nos permite identificar cuáles son los puntos de trabajo que vamos a tener. Can we mark this? Oh, it's your rock. Can we just make like a marker?
Exactly.
Yeah, it's exactly at 11. Okay, perfect.
Keep
Yeah.
waypoints right on.
Y, y lo interesante es que a veces este mapeo batimétrico no tiene la resolución suficiente para identificar este tipo de formaciones, así que ayer de hecho nos tomó por sorpresa descubrir un pequeño monte submarino en un sector que creímos que estaba únicamente cubierto por sedimento, y es justo lo mismo que nos sucedió en este momento. La batimetría lo que indicaba era que esto era un fondo de sedimento, y como pueden ver en realidad hay una estructura de roca. Esto que vemos en pantalla ahorita es una estrella de mar en una posición bastante interesante. Aquí vale la pena destacar que precisamente en sitios como estos, donde la gran mayoría está cubierto por sedimentos o por fondos blandos, cualquier lugar que haya con un sustrato un poco más firme, rocas, basaltos, alguna formación calcárea, se convierten como en pequeños oasis, donde entonces muchísimos organismos tienen la posibilidad de comenzar a sentarse, a colonizar, y vean que hay un cambio bastante drástico entre la fauna que habíamos visto en fondos de sedimentos y lo que estamos viendo en este momento. Maybe just take like a quick close-up of this, because I think we actually have to keep moving to the next waypoint. So
Yeah. You want another close-up of that?
yeah, and if it's possible with the lasers on.
Yeah, let's see how thick this
Yeah.
is.
Lasers on.
Oh, yeah, there are. Estamos utilizando los láser como una escala para tratar de estimar el tamaño de algunos de los organismos que tenemos en estas rocas. I think that's a sponge. Oh, sorry, it's just... Okay, sorry. Muchísimas gracias a las personas que nos acompañaron durante esta hora. Ahora le paso el mando o el liderazgo del buceo a mi colega Gustavo Ramírez, y me despido de ustedes. Pura vida. Thank you very much. Now I'm heading the microphone to my colleague Gustavo Ramírez. Has been a real pleasure to be with you. Aquí estamos marcando los que estamos ahorita justamente en el... No, I think we are just going to keep moving, yeah, and then probably try to reach halfway to the next point, so if we have sediment, we can try to do another heat measurement.
3, 4, 5 more waypoints to the summit, right?
Yeah.
Okay.
And the thing is like...
That was right at waypoint 11. So right there. And as you can see on our bathymetry, there's maybe the ghost of something there, but not anything super detailed. But we've still got a ways to go. We're headed ultimately to waypoint 16.
Good afternoon, everyone. My name is Gustavo Ramirez. I'm an assistant professor in microbial ecology and computational biology from the California State University in Los Angeles. we find ourselves about 150 or 60 kilometers away from the Costa Rican coast, exploring this incredible seafloor edifices, where almost 10 years ago, in very unusual behavior by octopuses was described here, and that was the main motivation for returning on board the research vessel Falcor 2 now, managed by the Schmidt Ocean Institute, for returning to these octopus gardens at the Costa Rican seafloor. Got a biosample there?
Sorry?
Is that something that we want?
No, the thing is like we are not sampling.
Okay.
We are just like on transit. We are just
Taking
sampling
photos.
wherever we
Okay.
are doing heat flow.
Okay.
So yeah,
All right.
that's what I thought. Like in the previous transit, and apparently it was like a star.
Our expedition's goal is to understand the rationale, I guess, ecological, scientific rationale behind a very peculiar observation. Again, first noted 10 years ago when exploring an outcrop that is an extrusion of the first layer of the crust here in Costa Rican waters, an outcrop called El Dorado. There's plenty of sites on this outcrop, which is a pile of rubble more or less at the seafloor, where hydrothermal fluids emanate into the relatively colder background deep sea water. At these sites specifically, where there was shimmering fluids slightly warmer than the background seawater, basically 10 to 12 degree fluids, emanating into 2 degree waters all around the deep ocean. At those sites specifically, there appeared to be congregations of octopus females, deep sea Pacific octopus females, that appear very interested in bare rock around the emanating fluids. It was very clear, again, 10 years ago, that these females were there to lay egg clutches and then brood them. The brooding behavior is quite picturesque. It's basically the females laying a clutch of eggs with a very hardy semen of approximately 6 to 12 eggs. As, again, this is a non-expert observation. This is just me counting them. And then subsequently, the females, after laying the eggs, appear to embrace their eggs in like this upside-down, inside-out hug that could last years, according to the experts on board. What was not fully determined when the site and the behavior of the octopuses was first discovered 10 years ago, what was not really clear as to whether or not the embryos that the thousands of embryos that these at least hundreds, if not thousands of octopus were brooding, if they were viable. So that was a major goal of this expedition, is it was to return to this one outcrop called El Dorado to determine the viability of the 1,000 of octopus embryos laden around the sites of hydrothermal discharge. During our first dive, that was 9 dives ago, I believe we're on our 10th one now. During our first dive, this Octopus Odyssey Expedition, the remotely operated submarine vehicle, whose camera angle you're seeing on your screen now, this ROV, remotely operated submarine vehicle SuBastian, returned to this site called El Dorado and found that, number one, that the octopus nurseries remained there. That was not known. So they were seen there 10 years ago, but we were able to confirm that they stay there for 10 years because we saw them. And even more importantly than that, we had a couple of close-ups of many egg clutches, and it was quite easy to see that there was very happy embryos within the eggs, more or less staring back at us. After a few minutes of being at the nursery site, we were completely elated to also see brand new hatchlings swimming right in front of our camera. So that's again, that was our first dive 9 dives ago, and that's been more or less one of the top highlights of our expedition. So we've been quite fortunate in terms of the luck that's required to persevere through the unknown, right? Because ultimately, we are here doing exploration more than anything else. To give the audience a little more background, this hydrothermal fluid venting that's observed seafloor features in this area, like Dorado, where the octopus females very purposefully appear to lay their egg clutches. This property is ultimately, of course, an emergent property of the physics of the ocean. So we're lucky to have on board a team of physicists with us, geophysicists, that are studying based on a series of very widespread, decades-long, and sophisticated temperature measurements at the sediments that blanket the seafloor around this seamount systems. They're able to study by generating very complex computational models the projected path of the fluid flow within the first layer of the bedrock that's on average hundreds of meters under mud in this part of the Pacific. Again, parts of that bedrock can protrude or rise up above the sediment layer, and if they rise up to a significant height, that's more or less what we would consider a seamount. Slight differences between seamounts and outcrops in this area drive or change the density of the water column, and ultimately drive the hydrothermal or thermal siphon driven circulation of deep seawater entering the crust into sediment cover aquifers underneath the bottom of the ocean that evolve the water thermically, geochemically, and microbiologically to eventual endpoints of their circulation flow paths, where this water emerges as warm, geochemically and microbiologically distinct hydrothermal fluids. Again, this very basic, basic yet extremely extensive physical emergent property, right? It's just the temperature that drives the movement of this water, appears to have incredibly important ecological repercussions that not only affect microbes that live in the immediate water surrounding the hydrothermal venting sites, but also appear to affect the lovely and extremely charismatic macrobiological life. And again, the best example of that macro life or large life forms is the majestic deep sea octopuses.
We're taking we're making a measurement. Yeah. Okay. Can we get an overlay of the heat probe? Thank you.
Okay, so just to briefly talk about what's going on at this point in our dive, I guess I'll give a little bit of context too. Like I said, our first dive explored a place that had been visited by another remotely operated vehicle close to a decade ago. After exploring that same outcrop called El Dorado, however, every other place that we visited, every other feature that we've explored with this with our ROV SuBastian at the seafloor has been completely new in terms of having never been visited by humankind in the past. So it's extremely exciting to be on a cruise where 90% of what we're doing here, 90% of the images that we're seeing and sharing with you all over the world, again, 90% of these images are the first time that humans lay eyes on these particular features. So definitely a cruise of discovery here and exploration. what you're seeing on your screens now is a couple of things going on. In the color shot, you saw the SuBastian robotic arm introduce what's basically a rod, a metal rod, not a true instrument, just a metal rod, into the seafloor. This rod is about a meter long or close to a meter long, and it's simply meant to tell the geophysicists that are in the control room here with me whether or not the sediment is soft enough and deep enough to introduce their fancier thermistor probe that takes extremely accurate temperature readings of different depths within, or at different depths within the sediment. So once that Excalibur, or again just meter long rod, is introduced into the seafloor and confirmed by the pilots here that there doesn't encounter like a layer of rock just beneath the mud, then that gives us a green light to then introduce the actual thermistor sophisticated, the more sophisticated thermistor piece of equipment, and that's what you see in the black and white camera footage going on now.
These data look amazing, by the way. Iris. Very clean. No vibration.
Nice. That was Professor Rachel Lauer from the University of Calgary in Canada, who's leading the geophysicists in this expedition, confirming that, I quote, "This data look amazing."
Lures are off. What are the? We're getting good dragger bugs. That's actually there. That's lures off. That's a little better. Yeah, lower sounds
I should highlight that we are an extremely interdisciplinary group here on board the research vessel Falcor. There are teams of geophysicists, as I just mentioned, geochemists, paleoceanographers, marine biologists with various specializations, and at least 3 or 4 microbial ecologists like myself. And yeah, we all get different roles in the control room. Sometimes some of us will narrate what's happening live. Other times, an even more important role is actually data logging. So every biological observation that we want to keep a timestamp on has to go through the logger. And it's quite noteworthy to highlight that our current logger, Mr. Robert Perrin from the University of Calgary, is a geophysicist trying to figure out what this biological sample that we're looking at is. So we've got plenty of help, but it's extremely humbling to take on this other roles. Yeah. So. Today we find ourselves exploring the vicinity, or the Piedmont really, coming up to the slope here, of a seafloor feature called Caballito, or putatively called Caballito. None of these names are official. That's an ongoing conversation with the science party here as to whether or not we want to take the first steps to officially name some of these features. So again, that's an ongoing conversation that we plan on further discussing in our subsequent lab meetings. Anyway, we are close to arriving at a relatively large seafloor feature called Caballito, or putatively called Caballito. This is after spending 2 days at a feature just east of where we're at now. That's again an unnamed feature that we're simply referring to as seafloor feature east of Caballito. We're there for 2 days, and in that feature, we're able to see some at least small areas of hydrothermal discharge as well, including some brooding octopus. The relationship between flow patterns throughout these outcrops and seamounts is, of course, ultimately determined by the physics in the crust underneath the sediments, and that is exactly the information that we are collecting now. This sediment temperature measurements will ultimately inform the geophysicists as to what's potentially happened with the fluid flow underneath the sediments and in the first layer of the lithosphere here. The geophysicists can then have some pretty sophisticated hypotheses as to where we should expect to find fluids coming out of the protruding bedrock, and then that's more or less the fluids emerging bring with them warmth, energy, and potentially unique microbiome that we believe, or we're hypothesizing, has profound influences for the ecology of this entire environment, or seamount environment. So we study it all here. Mathematics, physics, geochemistry, and biology. Can't have one without the other. Whether you like it or not. We don't do this because it's easy. We do this because there's no other way of doing this. Ask not what you can do for the seafloor. Wait, ask not what the seafloor can do for you. Ask what you can do for the seafloor.
Oh my god.
All the Kennedy quotes. Seems
Ooh, it's like a polychaete.
polychaete.
I wasn't quoting on that, but
Sure.
yeah, it's like, yeah, a worm. You've seen them like swimming in the water.
While waiting for the final thermal equilibration of the thermistor that's taking the very fancy, sophisticated temperature measurements at distinct depth intervals in the sediment here, we are free to basically pan our camera and just move around and have a very, very close-up view of all the benthic life all around us. So.
What's gonna happen?
Yeah. The tension builds as this centipede-looking critter gets pretty close to what appears to be the tentacles of another hidden invertebrate. Yeah, we're not sure what's going to happen here. We don't know how the day's going to go for our centipede-looking buddy here, but we're just looking right alongside you. Yeah, again, the tension in the room is, I guess, equivalent of seeing a National Geographic documentary, right, where the zebra is drinking at the water hole, with the croc eyes staring, right, breaking just over the the water level there. Okay, this zebra made it. Yeah. No, yeah, okay. All right. Happily galloping away.
And off it goes. We'll live to see another day.
Yeah. Fantastic. Anyway, I always like to highlight my biases as a microbial ecologist interested in microbial communities that live in the sediment. I rarely get a chance to focus on the macro life that's right at the interface here, like our what we're seeing right now. Every time I see mud, my mind immediately goes to the tens of millions of microbes and their extremely diverse activities that occur in every cubic centimeter of mud picked up from anywhere on Earth. So that's really where my mind usually goes, the microbial activities of the mud. But it's certainly interesting to sit here and look at the macro biological life, the charismatic macro life here.
Our zebra has appeared to slipped into a hole.
Yeah. It's back into its comfy abode.
See if I can find some more drama unfolding.
Yeah, yeah. We're on the search for benthic drama here. So let's see what else is around here.
Benthic drama.
Yeah, benthic safari. Wow, those starring all around.
Yeah.
Yeah. It's hard to tell if that's an offensive or defensive strategy, right?
Yeah.
Yeah.
I said earlier, it's like my kids at the beach, you know, they cover themselves. Maybe it's just fun.
Maybe it's just fun, or maybe there's skates that swim by occasionally. Yeah.
What do they feed on?
We have to get Oda over here. Yeah. If you can see it, I know nothing about it. Yeah.
No, I just
Those small orifices at the seafloor appear to be burrowing sites for invertebrates. Wow, that's pretty interesting. A what? Wow, okay. I can't even pronounce it. Say it again. Xenophytophore?
You're taking the advice.
Yeah.
Thrusters caught me and kicked those up right there. Subsea tornado.
Ah, nice. Okay, we're all done with the measurement? Cool.
Oh, we are?
I don't know. I'm asking. Yeah.
What's that?
Are we done? No.
100 seconds.
Okay.
Could you say something to them? I just have like
About a minute and a half here to go.
So I think a lot of that optimistic water collecting. And I reached out to Barry Browse about it. And we're just having a conversation, but I wanted to make sure that we can confirm you and what they said that it's okay to do optimistic collecting at J-Rock. So
All samples we collect.
Again, we are en route to eventually summit previously unexplored seamount, putatively named Caballito, or Little Horse.
So, two questions that can you collect them and then discuss it with her, like, is it okay?
We have no clue as to what kind of hydrothermal activity we'll see there, or if we'll see anything at all. But again, the best way to develop a hypothesis or to know something a priori before showing up in person is to do these temperature transects that which basically involves inserting a series of thermistors on a metal rod that collect that are very sensitive thermometers. They're able to tell us about the rate of heat flow that's starting at the crust and emanating through or moving through the sediment layer here.
The reason I'm asking is I want us to speed up.
If heat flow was indeed detected at this area, we would expect that the aquifer beneath would be relatively warm, and that would imply lower density fluids that are potentially emanating at when we get closer to the summit of this seamount. Again, putatively called Caballito, El Caballito.
Let's probe up.
On the boosters, whatever you guys did, the last couple of measurements, you tell your friends, just like whatever positioning, because it was like, usually there's this shatter,
A little bit is I'm trying to stay in the 60s, my range from the vessel, and a little bit is the angle of this slope is allowing that side to go a little bit deeper.
Right, okay, that's what.
I don't know which one it is or both, but I'll let them.
Yeah, no, I just, thank you, sorry.
Did she tell you?
Yep.
Okay.
So if we're done with heat
Yeah,
probe,
we're done.
We're good?
Done with
Okay.
heat flow.
we can probably stow this guy.
So we are in between the 11th and 12th waypoints for this dive, with the scheduled total of 16 waypoints, the 16th waypoint being placed at the summit point of this previously unexplored seamount called Caballito, or putatively called Caballito. Our geophysicists appear to be very satisfied with the many temperature measurements that have been taken on this sediment so far, or on this dive so far. So we will speed up the pace here and be on our way to the summit. Again, my name is Gustavo Ramirez. I am an assistant professor of microbial ecology and computational biology from the California State University in Los Angeles. My primary focus during this expedition is understanding the nature of rock and microbe interactions, specifically microbes that emerge into the deep ocean from openings at the seamounts where hydrothermal fluids vent. So again, those hydrothermal fluids they have a different temperature than the surrounding ocean. That's why they look shimmery. They have a different geochemical profile than the surrounding ocean. And that's why they can sometimes precipitate things like metals. And they also have a different microbiological signature, different communities, distinctly different communities than the background seawater. So I am interested in seeing how the microbes that come out of the crust, that are part of these hydrothermal fluids, how do they interact with the non-living parts of the immediate environment in the seamount, that is rocks and sediment around places of venting. Again, we are on our way to waypoint 12 out of a total of 16 waypoints scheduled into this dive. The 16th waypoint would be at the topographical maximum in this area, the summit. That is the summit of a seafloor feature putatively called Caballito, a seamount never before seen by human eyes until today's dive. It's pretty hard to convey the level of excitement that this pure and applied exploration can bring to a group of scientists out here. But again, every image that we see and share with you live through YouTube represents an environment, again, never before seen by human eyes. And I never get tired of highlighting that. It's extremely important to note that. We are very fortunate.
Instead of going down at it, like pitch out, shoulder wave down, so you're flatter. So when you let it go, you still paddle. You have a little bit more. You're not pinned against the box.
That's a good point.
It's sort of difficult. You'll have to put the tip in there because once you change that whole picture is artificial. So
I think I have one of those right here. Is that this?
We've seen lots of them. I don't think it's on the bottom.
Yeah, so I can't get that. Just get that box. Yeah, I couldn't have a transect at least.
Yeah, so you get a bit of angle there. Just slowly open your drum. You're all right.
Bienvenidos a todos los hispanos parlantes que nos sintonizan durante este buceo. Me llamo Gustavo Ramírez y soy un profesor en la Universidad Cal State en Los Ángeles, Estados Unidos, y me encuentro en el barco de investigación, el buque de investigación Falkor, que es parte del Schmidt Ocean Institute, y estamos a más o menos 3,000 metros de profundidad explorando una zona en el Pacífico costarricense, una zona de montañas submarinas. En estas montañas se ha documentado una actividad de pulpos profundos que nunca se había documentado antes. Básicamente lo que se pudo ver aquí hace 10 años aproximadamente fue una colonia de hembras pulpo que específicamente dejaban sus huevecillos sobre rocas expuestas debido a fluidos hidrotérmicos que salen en piedras en medio de montañas submarinas. Este fenómeno es lo que venimos a investigar primordialmente durante esta expedición que se llama la Odisea del Pulpo, o Octopus Odyssey. Pero bueno, para investigarlo a toda su profundidad se necesita saber bastante acerca de la geoquímica, geofísica y microbiología que ocurre en el sublecho marino. Bueno, me gustaría reiterar que cualquier pregunta que exista acerca de este buceo, hay un gran número de hispanoparlantes que somos parte de la tripulación científica de esta expedición. La mitad de la expedición, la mitad de los científicos en esta expedición son colegas costarricenses que nos acompañan. Es importantísimo para los costarricenses estudiar el lecho marino, ya que el 90% de su territorio nacional es exactamente este medio ambiente que estamos viendo aquí, que es el lecho marino profundo. Voy a resumir con mi narración en inglés, pero cualquier pregunta que exista en el español, yo o cualquiera uno de mis colegas hispanoparlantes aquí en el buque la podemos responder. Really? Could that be a rope?
That does look more like rope, doesn't it?
Yeah. Bummer, we can't collect. We can't collect, right? We're on a... Yeah, we got to make it to the summit, so it's unfortunate. It's an observation.
Yeah.
Well, it could have been a gigantic fecal pellet too, you know?
That was my first thought, was
Yeah.
like, wow, some really twisty echinoderm.
Yeah.
Produced that. Very talented. Breaking them
So we saw an irregular structure at the seafloor here that looked more or less like a rope. We can't confirm that, but I guess other similar structures, yet of much smaller size, all around us, we know to be fecal pellets. So those, for example, what you're seeing there on the right side of your screen there, we're flying right over it, is what appears to be a fecal pellet or fecal spiral excreted by sea slugs usually, right?
Mm-hmm. A combination of things. I didn't see the one you're talking about,
Okay.
but the acorn worms are producing them, the sea cucumbers are producing them, so the larger ones that are kind of spiraling could be either of those two things.
Okay, so yeah, not surprisingly, there's plenty of remnants of animal excretion on the seafloor here, and yeah, we're not sure what that structure was. Probably 3 times or 4 times larger than the largest animal excretion or animal excretion pellet that we've seen here. So we're not sure. Could have been anthropogenic in nature. We just don't know, and we can't stop at this moment because we want to make sure that we hit all of our waypoints and summit this previously unexplored seamount. Saludos, tocayo, un abrazo a todo Venezuela y gracias por acompañarnos. We have now passed our 12th waypoint. We're on our way to the 13th, and it's about a waypoint or 2 to go before we see the highest rate of elevation change that we expect for this dive. So we will hit a seamount, a pretty big structure, quite soon. And again, what we'll see there in terms of hydrothermal venting and the generally associated wonderful macrofauna like octopus and skate nurseries that we've seen before too at other summits, we just don't know because this is again a completely unexplored site. abyssal plains, just like the ones that we're seeing on the screen here, cover much of the Earth's ocean floor, oceanic surface. An interesting point to bring up at this point, however, is that this structure that we're about to encounter, this seamount putatively named Caballito, when you look at it on a map of the region, all of these seamounts and outcrops are, you almost have to squint. They look like very small pimples on across the seafloor. However, they're everywhere. So a very interesting thing that or fact that I came across in the literature just a few days ago is that despite the seamounts being again extremely small relative to the giant plains of sediment all around them, if they were to be combined all together in one place on Earth, they add up to a surface area of approximately 30 million kilometers. So I didn't know how to contextualize that, so I basically Googled what 30 million kilometers was, and it turns out that it's the size of the continent of Africa. So what we're doing here is learning a little more, one football field at a time, basically, with this very focused dives of something that's happening on an environment that's, in terms of its surface area on the planet, is equivalent to the continent of Africa. So that's the level of importance that these small, relatively small structures at the seafloor collectively have on very important biogeochemical processes, potentially, at the planetary scale. Couple more twisties here. Yeah.
Floods seem to do the damage. I think it has to do with like altitude too. So if I'm lower now, if I come up, I think I'll see it. Yeah, see it. There it is, yeah. So it's like everything else over right.
There appears to be a change in topography and geological structures coming up here, and as you can see, as soon as the geology changes, the biology changes also. This is yeah, we kind of take this for granted, but this was not something that was explicitly studied until the round the world expeditions of the Prussian scientist Alexander von Humboldt. So it's quite remarkable. Just one little break in the sediment is enough to have a little local explosion of diversity. And just like that, we're past that little feature and we're back to this abyss of magnificent desolation, as the astronauts describe the surface of the moon, the Apollo astronauts.
ROV bridge, are we going to be coming and stop here, or are we keep going?
Good. Yeah. You can jump. Yeah, jump in. We are okay, getting to waypoint 13, so we did 12.
We already did 12?
Yeah.
Okay. And so we have to go to the summit and that's it?
Yeah, and like I don't think they're official transects, but I'll check with Beth right now. Okay. I think that we just have to summit. Don't stop for anything. Keep going. Gas it once we get to the summit. If there's time, we do other stuff, but it's just we gotta survey the area.
And normally summit will be lava flows and
It's already changing. And the slope is going to change real good right now.
Yeah.
So it's just a matter of time before we start going up the slope.
Okay.
Yeah. what happened before, mostly heat flow. So again, brand new site, unexplored, so it's they needed the heat flow transect, so I think they did 4 or 5. Apparently very good ones.
Es diferente. Eso no se desarrolló, ¿no? Pero si veo la licencia de tu automóvil o la placa, para siempre, en 10 años hablamos de eso.
Again, to everyone in the audience, it's been a pleasure hanging out with you and narrating the beautiful sights of the seafloor as we're exploring again a feature here, a seamount never seen before by human eyes. So it's an incredible privilege to be sitting here in the control room of the ROV that's doing this really cool high-level exploration of a very important part of our planet. I will be signing off and turning over the microphone to my colleague, Maria Sandoval, a professor of paleoceanography at the University of Costa Rica. So, Maria, take it away.
Thank you. Hello everyone, this is Maria Sandoval from University of Costa Rica. I work at the geological department at the university. So if you have any geological questions about it, just let me know in the chat. Hola a todos y a todas, muy buenas tardes. Les habla María Isabel Sandoval de la Escuela Centroamericana de Geología de la Universidad de Costa Rica. Y voy a estar con ustedes esta hora, por lo tanto pueden realizar sus consultas en el chat. Voy a estar narrando principalmente en español esta hora, por lo tanto podemos conversar un poco sobre lo interesantes, este interesante sitio que hemos estado visitando el día de hoy. Les comento, este lugar se llama, es un afloramiento rocoso llamado el Caballito, o pues esto no es un nombre formal, sino fue descrito así en unos estudios antiguos, por lo tanto se sigue usando este tipo de nombres formales en toda esta región que estamos entre 150 a 200 kilómetros de la costa costarricense en el Pacífico Norte. Estos pequeños montículos y seamounts asociados provenientes de la dorsal del Pacífico, con edades aproximadas de 20 millones de años en adelante. Y este lugar no han sido oficialmente nombrados, por lo que cuando usamos los nombres acá son principalmente nombres que se han utilizado pero no se han oficializado, ya que esto amerita un estudio y una solicitud formal. Aquí tenemos un pepino de mar bastante distinto a lo que estamos viendo. Estos pepinos de mar viven principalmente acá en el sedimento, consumiendo y nutriéndose del sedimento también. También podemos ver excrementos casi siempre en estas partes más planas, comparado a lo que vimos, por ejemplo, o hemos visto en otros sitios como El Dorado o más temprano que vimos lavas, por ejemplo. Ahí en este caso tenemos menos, la abundancia de estos organismos es menor. Todo lo que estas estos como rollitos que vemos ahí son este tipo de excrementos de lo que son los pepinos de mar. Aquí vemos un erizo muy colorido, un erizo irregular, también familia de los equinodermos. Toda esta familia es bastante abundante en estas planicies abisales. Les recuerdo que estamos a 2.830 metros y estamos principalmente observando ofiuroideas, equinodermos, erizos. También hemos visto varios, ¿verdad? Distintos tamaños y colores. Just to let you know that here in that place, in this abyssal plain that we have been observing, principally is the group of echinoderms. We have sea cucumbers, brittle stars, some urchins of different colors, and they are very abundant on these abyssal plains compared to the places that we have seen before in the morning and that were more rocky. Hemos estado observando también algunos parches o variaciones en el sedimento, donde hay en la parte más café tenemos un poco más de materia orgánica y arcillas, y algunas partes más blancas hemos podido ver también aflorando en algunos escarpes bastante interesantes acá en este sitio. Estas rocas blancas que consisten principalmente de huces o barros calcáreos que se forman por la acumulación de organismos como el fitoplancton, que al morir caen al fondo oceánico y van acumulando estos esqueletos. También hemos podido observar por medio del estudio de los sedimentos y de los testigos de push core, los testigos de sedimento que hemos sacado en esta expedición. En estas láminas observamos por medio del microscopio lo que es la composición de estos sedimentos. Esos sedimentos, como les dije, principalmente contienen organismos o lo que llamamos lluvia pelágica, o también se llama nieve marina, y es esta acumulación de miles a millones de años de esta nieve marina en el fondo oceánico. So those white spots that you can see at the bottom we call it calcareous ooze, and they are formed by the accumulation of the foraminifera and coccolith and part of the marine snow. So they accumulate at the bottom during thousands and million years. So we have plenty of brittle stars in that area. Also these funny purple sea cucumbers that are, I don't have the genus here, but we have observed a lot of these organisms in these abyssal plains. Bueno, al lado mío, observando y anotando todas las observaciones biológicas y geológicas, tenemos al estudiante de doctorado, el ROV de la Universidad de Calgary. Rob Perrin de la Universidad de Calgary. He estado acá durante 2 horas, lo que es tomando nota de todos estos sitios y observaciones que hacemos en un programa especial. Les comento que en general cuando llegamos, el robot llega a lo que es la superficie marina, se inicia los videos y la transmisión, y a partir de ahí todo esto queda en programas, en anotaciones, y podemos también a veces hacer algunas aproximaciones a algunos organismos, ya que no todos los organismos se pueden colectar, ¿verdad? Algunos organismos simplemente los observamos y con esto al menos tenemos una base de datos de lo que es toda la diversidad marina a estas profundidades. Les comento, esta es la primera vez que estamos en este sitio conocido como el Caballito. Nunca antes nadie había estado en este sector, así que es la primera vez. Así que aprovechamos estas imágenes por primera vez en transmisión, tanto para nosotros como para ustedes. Así que es un placer gozar de poder ver este sitio por primera vez y totalmente libre, algo que es muy valioso de esta organización del Schmidt Ocean, que permite que todos los videos y lo que vamos viendo no solo queden para la ciencia, sino queden para la divulgación de todos estos sitios. Aquí vamos hacia lo que es vamos en nuestro camino hacia la cima de este afloramiento rocoso que se llama el Caballito. En esta parte estamos en la parte plana, o en lo que llamamos el fondo oceánico, planicie abisal. The objective for today is to visit 16 points. Already we are in our path to waypoint 12. So we having so nice images from the abyssal part, but probably we'll get having more rocks to the summit, and to get some more measurements there and more observations. Each waypoint is divided in 100 meters. En lo que son estas zonas abisales se observan unos organismos bastante grandes, a pesar de las profundidades y de las condiciones tan especiales para vivir acá. La limitante de oxígeno, la limitante de presiones y demás, Son sitios muy particulares y vean la gran cantidad de fauna que tenemos, ¿verdad? Peces muy particulares, ¿verdad? Con formas muy particulares. Inclusive también algo que me llama mucho la atención es los colores que hay acá en este fondo profundo, o esta Costa Rica desconocida. Les recuerdo que esta luz que estamos viendo es falsa, es la luz que tiene el robot, ¿verdad? Es proyectada, pero aquí no hay nada de luz. La luz que a veces se puede observar es de la bioluminiscencia de algunos organismos, pero esta no es posible captarla con nuestro equipo. Esas acumulaciones de sedimento como en línea que se ven, donde se ve una parte más blanca y un tipo de montículos, estos son bioturbación que realizan algunos organismos, no sé exactamente qué organismos son, pero todo esto es bioturbación. All these patches, round patches that we see here, that are created for organisms. I don't know exactly which organism, but we call bioturbation. Les comento que en esta expedición tenemos diferentes investigadores de distintas áreas, por lo tanto es un equipo interdisciplinario. A bordo tenemos también 2 artistas, cosa que ha sido una gran oportunidad tanto para nosotros como para ellos, ya que inspirados en todas estas imágenes, en las observaciones, en los organismos que se han colectado, se basan para realizar imágenes espectaculares, y pueden seguir las páginas de ambos artistas. Ya ahorita les digo sus nombres para acá fijarme en Instagram. Bueno, tenemos la página de Michelle Droch en Instagram y también a Carlos Hiller. Ambos pueden buscarlos. A Michelle lo pueden buscar como michelle.droch.studio y a Carlos lo pueden buscar como Carlos Hiller. con H, artist. Ambos han hecho un gran trabajo a bordo y las imágenes son compartidas día a día, así que ha sido una gran experiencia tener este tipo de equipo, ¿verdad? Tan variado. Tenemos investigadores desde las áreas de microbiología, geología, geofísica, hidrogeología, biología principalmente, pero dentro de biólogos tenemos también distintas especialidades de distintos organismos o que estudian distintos organismos. Por lo tanto, las conversaciones se vuelven generalmente muy interesantes, narrando y tratando de conocer distintas especies. Todos los días conocemos distintas especies, inclusive grupos que tal vez alguno u otro no conocíamos. Y esto ha sido una experiencia muy interesante también, y por supuesto también desde el punto geológico ha sido también muy interesante poder hablar y dar a conocer los trabajos que hacemos desde la geología a nuestros colegas. Aquí tenemos una estrella de mar que vive ahí sobre este tipo de octocoral. To let you know that we have a multidisciplinary group here, but also on board there are 2 artists that you can follow the web page on Instagram. I will let you know the first one is michelle.droch.studio, and the other one is Carlos Hiller, artist. You can follow their paints and their inspiration from this expedition, or from all this imagery that we have kept during all these days through their paintings. Aquí tenemos uno de estos gusanos spaghetti worm.
What is that on? It's Joshua Amazon. Oh. Do you know what that is?
Yes, it's a spaghetti worm.
Spaghetti worm?
Yeah. It looks like that. I'm checking just with my colleagues. Yeah. 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 I think I will ask in the chat, but I'm almost sure. Collect, she said. Okay.
Collect.
Yes, please. Cool.
Yeah, I'll start going down now.
Este es un gusano espagueti. Son muy raros de ver y en esta expedición hemos tenido la oportunidad de ver unas 3 especímenes de estos. Por supuesto que habrá muchos más, pero a veces no estamos tan cerca del fondo como para observarlos. Así que aprovechen estas imágenes. Este organismo lo vamos a colectar. Is the first angle?
Where would you like it to go?
In Coral Quiver one.
Okay, because it's
Is it big one?
how much of it is it? Will it all come up as one, or is it like a whole bunch of little ones?
I think it's just one.
Okay.
Like a bowl.
What size is it?
I know we can.
I'll
Yeah,
turn the
please.
lasers
Yes,
on.
please. Yes.
Could be very big.
Yeah. Oh, maybe not. Maybe 10 centimeters.
So yeah, so roughly the lasers are the opening of a coral quiver. So I mean, we should be able to get in there as long as it's not like.
Okay.
I've never seen where they, so I don't know what to expect when we go to touch it.
Maybe contracts, so it will be easy to collect the whole thing.
Yeah.
We could give it a little bit of a boot and see what it does.
Okay.
See if
Yes.
it cuts it.
Okay. Yes, please. Yeah.
A boot.
Yeah.
Science boot.
So we will try to collect this organism. This is spaghetti worm. They have been also studied and observed in other areas in the deep ocean. Yes. Mm-hmm.
We will take it maybe with the scoop.
Wow. But then taking the scoop and getting it to fall into the. We have open quiver wells. Are all the quivers open? Yeah. They're all open.
Yeah.
Go down a little bit deeper when you go to the pool, or just sit around with that top spot. We could also try suction sampler. I don't know if it'll come up with the suction.
Yeah, could be. Yeah. I think I prefer to take all I mean, not in parts, but all the specimen if we can.
Yeah, I didn't know if maybe the suction sampler would like come up like that and then we would.
Yeah. Yeah.
Or is it all the individual, see what the ones that shrink up?
Yeah.
This is a very strange image. Okay. We got the parts to come out?
grab onto you. See that? So it does kind of break apart. You see that one little piece that's on the tip of the arm?
Yeah.
So it does seem like it's a little fragile.
Yeah.
Okay, that's,
Aquí estamos tratando de conocer cuál será un poco la reacción o un poco también la consistencia de este organismo, ya que nunca ha sido colectado. Por lo tanto, estamos tratando de ver con los diferentes equipos que cuenta el robot. Aquí vemos un pez bastante curioso. He wants to know what happened. Parte de las colecciones que se realizan durante la expedición son llevadas al Museo de Zoología de la Universidad de Costa Rica y también a los diferentes museos a nivel internacional donde los investigadores que están colectando acá y son especialistas en el área también se llevan a distintos museos en el mundo, principalmente Estados Unidos, para ser analizados tanto desde la parte genética como la parte taxonómica. Me indican acá que este organismo había sido colectado en una expedición en el 2019 y parecía ser una especie nueva de este tipo de gusanos espagueti. Apparently, the size of the organism is more than 10 centimeters. Could be 20, maybe. That's cool, right?
It's coming off my vertical thruster. See up there?
Yes.
So that corkscrew comes all the way up and then comes down.
Hmm.
So what you want to do is come up and over and get almost like a straight down grab on that bottom. Yes. So like if you can like keep your grab like that and just like start moving your arm into that position, that'll be the best grab for being able to fall down.
Bueno, esas son algunas de las herramientas con que cuenta el robot para poder colectar. A veces tenemos que probar varias de estas herramientas. Se toma bastante tiempo para tomar el espécimen de la mejor manera. Por lo tanto, ustedes vieron que probamos primero con lo que es la tijera, pero la idea es tener el espécimen completo y no en partes. Así que intentaremos con esta cuchara. Mm-hmm. So here we will try to collect the spaghetti worm that was also observed in a further expedition in 2019 and apparently was a new species, so it would be interesting to analyze this specimen here. So we are trying to get the best way to collect it. With the lasers on, we have an idea what is the size of this organism. It's apparently maybe 10 to 15 centimeters.
Oh, it's starting to go.
Mm-hmm.
Whoa.
Oosh. Well, it's bigger than we thought.
You might get them though.
Oh my God. Wow.
Oh.
Mm. It's huge.
Yeah, if he's trying to lift off.
Okay.
Is there any other way you want to simplify?
Oh, he's on the porch now.
Suction or bio box? Anything like, will a bio box work for you?
Is it right there? Where is
Yeah,
it?
he's right here in the front porch. But like, would a bio box work for you?
I will ask Beth.
We're trying to get him in the.
They're all
Okay.
empty.
Yeah.
Let me try to get him in the jar.
Okay.
Yeah, it could go in a bio box.
Bio box could be?
Yeah, it could go in
Okay.
a bio box because they're reserved for octopus and we're unlikely to.
Oh, okay. Okay. Thank you. Okay.
Right, just go ahead and get your arm in the safe spot, off to the right. One of the bio box 3.
Yeah. Bueno, como vimos, era mucho más grande de lo que suponíamos. Así que intentaremos, bueno, ya parece que dentro de alguno de los equipos que tenemos acá en el robot. Así que se tratará de capturar para poder ingresarlo a una de estas cajas de organismos. Ahí está. Ok. Perfecto. That was
Wow.
great. Okay, so
Don't like that.
That was a
yeah.
organism.
Okay, Biobox 3A, yeah.
What is it for you? Just so you know, I'm going to be working my way down.
Yes. We were lucky, yes.
I can't keep doing it and just give up. You can go ahead and start.
Oof.
So here we will continue moving to the Caballito outcrop. And we are trying to get the summit. Bueno, ahora después de haber colectado este organismo, seguiremos hacia el camino que nos lleva por los diferentes puntos que tenemos hoy como objetivos, que son 16 puntos. Estos puntos están divididos casi en metros. En la mañana estuvimos principalmente haciendo algunas medidas de calor. Este calor pues aparentemente se mantiene bastante bajo, por lo que se supone que es un sitio de recarga y no como el sitio que vimos ayer, que supondríamos es un sitio al oeste de Caballito. Teníamos ayer estuvimos viendo algunas capturas de pulpos y un poco de medidas de temperatura mayores que nos indicaban un sitio de descarga. Eso quiere decir que, como ya lo hemos comentado, en uno de los sitios, por ejemplo en este Caballito, tendríamos el agua que ingresa a la corteza por medio de estos montículos de lavas, y esto se mueve, el flujo de agua va a moverse y tomar calor de la corteza y salir en sitios donde, como el sitio que estuvimos ayer o como el Dorado, que el agua caliente o aún más caliente que el agua circundante va a salir. Entonces estamos teniendo como acumulaciones, ¿verdad? O tipo seamounts, donde son las estructuras más grandes, es las que se creen que son de recarga, y las estructuras más pequeñas son sitios de descarga, y evidentemente donde hemos tenido la presencia de estos maravillosos organismos que son los pulpos anidando y incubando sus huevos. Aparentemente el calor es lo que los llama a incubar ahí. También se supone que favorece lo que es tener las rocas limpias, ¿verdad? Al salir el agua, las rocas se limpian, ¿verdad? Y del sedimento que cae o que se acumula en estos sitios, generalmente la lava estaba bastante limpia, y los pulpos, estos lugares también les gustan para pegar como un tipo de moco que tienen, o un material que utilizan para pegar los huevos y poder ahí protegerlos. ¿Arriba en qué sentido? Bueno, aquí tenemos lo que es una araña marina, que es un organismo llamado pignogónido. Yes, yes. Here we have a crab with these anemones. And also we saw a pycnogonid, yes, a sea spider to the left. Estos son un tipo de cangrejos que viven con sus tipo de anémonas, cargando estas anémonas, y son como tendríamos en superficie o en las aguas someras generalmente los cangrejos ermitaños. Pues aquí tenemos cangrejos ermitaños, pero que utilizan este tipo de anémonas. ¿Qué son anémonas? ¿Son anémonas o son anémonas? Sí. Okay. Sí, son un tipo de anémonas que crecen en su caparazón. Uh-huh.
Okay, it says Jorge wants this crab.
Crab has wanted for back.
I'm just gonna back up just a little bit, I'll keep an eye on him, just to see what this other, thing of nightmare is off on the right.
No, yo no.
That's what it looks like, yeah, they're just really spread out.
Yeah, so cool.
Jorge wants. Aquí tenemos al pycnogónido.
Get into a suction sampler or coral quiver?
Suction sampler is a negative.
Negative. Okay,
It's
coral quiver.
much bigger.
Coral quiver? Yeah.
Coral quiver, okay, that's going to be an interesting game. All right, so.
Yeah.
Because he can.
The crab, yeah.
Oh, I see.
But I mean, we could potentially maybe use the suction sampler to.
Yeah, does it come and go? Like, can you reverse it?
You can't reverse it, but we could probably capture it with a suction sampler.
Yeah.
And then get them over coral quiver, and then put them in the coral quiver and turn the suction off.
He's on the move.
Yeah, he's going real fast.
Good job. We get an all stop on that move and do a push.
There was a question about whether or not we use like pressurized containers or aquaria to hold these animals. And no, we're bringing these up in non-pressurized containers. They don't experience the dramatic explosive barotrauma that something like a fish with a gas-filled swim bladder would. And there are hyperbaric collection vessels that are developed, including ones that even can be taken by divers down simply, 100 meters to collect some mesophotic fish. But ours aren't pressurized or anything like that.
Bueno, acá la idea sería poder colectar este organismo, este cangrejo ermitaño en la profundidad con sus anémonas. Vemos que va a ser como un poco complicado la captura del organismo, ya que está caminando y caminando, pero bueno, eso se intentará. Acá parece ser...
You're going to want to get that nozzle pointing down, because you're going to want to get that, and then lean on the side for the suction. That makes sense? Yeah. Just go ahead, just get in that position. I'm just going to keep on in case you don't take off. Simply. Pretty much just straight up and down.
Este que está acá blanco es el brosoario que me dijiste, pero para que lo veas. Es que no ya lo quitaron. Sí, muy lindo. Sí, sí, sí. Ajá, ajá.
Go ahead and elbow out and touch. And you're going to need to go a little bit like that.
Bueno, mientras van a tratar de realizar la colecta de este cangrejo por acá, les comento que pueden seguirlo o ingresar a la página del Schmidt Ocean y leer los blogs de esta expedición de parte de tanto de los científicos y científicas como de los artistas que estamos en la expedición, contando experiencias acerca tanto de la expedición como de los estudios que se están realizando en general.
exactly how aliens get people.
Nice. Mm-hmm. Good catch.
Go slowly start working your way to the front porch. Make it in there. You won't make it all the way. Okay, good. Keep coming in.
Cork with that one? Yeah, I think so.
All right, you see how much room you got over here with the camera? Don't shoulder left anymore. You're gonna have to rotate your wrist. Actually, shoulder right and let the porch out.
Estos organismos, este cangrejo, tiene algunas relaciones simbióticas con lo que son las anémonas o antidos, organismos que forman parte del filo Unidaria, y con estos se protegen, con estos organismos que crecen en su caparazón.
That's fine. Find that now the arms aren't as big as they look. Just get that nozzle right inside that hole now. Cut the suction.
Usually these crabs have this symbiotic relationship with anemones or zoanthids, part of the Nigerian phyla.
So you're gonna let go of the, if you need to do one of two things, I can get the port arm ready and put on the port arm, or you can just open your jaws, grab it, put it on, and make sure it goes down. That makes sense? Okay, yeah, so you got it, or you, I think you got it. All right, so legitimately, you got that plan, you're gonna open your jaws, come for that quiver top, and then go right on top of it. Getting it slammed down and completely sealed, not the primer, it's just on top.
Aquí los pilotos, que son bastante ágiles, planean lo que es la mejor colecta sin dañar el espécimen. Por lo tanto, a veces se dura mientras planean cómo poder realizar la mejor colecta. Recordemos que las colectas aquí pues son limitadas también por la logística, el tiempo que tenemos por día con el submarino bajo el agua, y por todos los otros objetivos que tenemos de muestreo. No simplemente se muestrean animales, se muestrean también testigos de sedimentos, se muestrean o se toman datos de temperatura, se muestrean agua, datos para análisis geoquímico, se muestrean también rocas, y todo esto lleva su tiempo y su dedicación, por lo que las colectas, como les mencionábamos, no son caprichosas, sino las colectas tienen su objetivo de investigación y de preservación también.
No, he's just waiting.
The specimen collections takes their time. So usually these collections or organism collections it has to be well informed, well coordinated for the pilots and with the scientists too. So usually we take the time to get the best collection, and especially that the animal get well preserved when arrive, and also to the quiver, and also to here when it coming out to the surface. So that's usually takes time to get the organism. Yeah.
He's starting to move a little bit. I'm just going to keep doing what I'm doing.
Yeah. Wow.
Hopefully he acts the same way when he's inside. I would really appreciate that. Give little wiggles as you slowly come up. No, don't come all the way out. Well, see if maybe he wants to go down this. It's almost out of there.
Algunos organismos, como vemos, son mucho más complicados de capturar. En el caso de este cangrejo, pues está bastante adherido, tratando de quedarse dentro del tubo. Así que está complicando un poco la salida de sí mismo hacia el otro tubo de colecta. Waypoint 15, waypoint 14, right?
Yeah, I think we got past.
Even the 15. Yeah, I think even.
Yeah, we've covered it.
Yeah.
So I think we can just go right from that. Keep that.
Yeah. Sí, por la facilidad que se tuvo capturando al spaghetti worm, en este caso se complicó mucho más la colecta del cangrejo. Un organismo bastante astuto.
As you can see, it does take a decent amount of patience with some of these collections. Patience and coordination.
you can enjoy and read the blogs that we have in the Schmidt Ocean page from this expedition, Octopus Odyssey. Yes, I think. Okay.
I try to split that eye with the jaws open. That way you can get on either side of it. And then just push down with nice.
Bueno, fue una captura un poco complicada, sobre todo un poco bloqueada, diría yo. Así que ahora ya se logró colectar este organismo satisfactoriamente y podremos seguir con nuestro camino hacia la parte,
Well
la
done,
última
pilots.
parte. Yeah. Good job. Sí. Bueno, entonces me despido de ustedes y les dejo la narración de nuestro colega, el compañero Sergio Cambronero del Departamento de Física de la Universidad Nacional de Costa Rica. Bye everyone. So I leave you with Sergio Cambronero from National University of Costa Rica.
All right, got a little bit of a watch handover happening here with the scientists.
Thank you very much to my colleague Maria from Universidad de Costa Rica. My name is Sergio Cambronero and I will be joining you for the next hour. This dive, we're in the summit of this seamount that is called Caballito, translate to little horse, and we're going to explore around. And for the next hour and towards the end of our dive, we're gonna be really paying special attention to any venting signs or shimmering water. Así que muchísimas gracias a mi colega María Sandoval de la Universidad de Costa Rica. Mi nombre es Sergio Cambronero y los voy a estar acompañando por la siguiente hora en este buceo, el número 11 de nuestra expedición. Estamos en un monte submarino llamado Caballito, explorando. Ya si se están conectando o recientemente incorporándose a este buceo, hemos estado haciendo aproximadamente 15 puntos de muestreo con nuestro sensor de temperatura para medir el flujo de calor. Pero ya una vez que estemos aquí, a donde estamos en este momento, que es la cima del monte, vamos a estar meramente dedicados a explorar por alguna evidencia de fluido hidrotermal o de este tipo de agua que se puede ver que a veces sale de la corteza oceánica. So thanks everyone who's tuning in. We are currently almost at 2,800 meters depth. This is our 11th dive of this expedition at Caballito, and right now we're gonna focus on any evidence of venting or shimmering water. So the next hour will be dedicated just to explore around. So bear with us and stay tuned as we discover and explore this never explored before seamount called Caballito. So I will also like to thank my colleagues here in the science party that joined me in the control room. Dr. Holly Lutz from Scripps Research and Dr. Odalis Cabridi, which is the octocoral expert on board. And we're gonna keep exploring, as I was telling you, while we take a close up at this beautiful shrimp. We have been seeing many shrimps of many different types in this expedition. So they are common bottom dwellers, and sometimes you would see them on top of the rocks just wandering around, some other times displaying these beautiful swimming patterns and behavior. We see some brittle action. Okay, it's very active in the afternoon here. It's almost 4:10 p.m. here in Costa Rica time, and I hope you're enjoying this live stream. Currently being live streamed from the offshore Pacific coast of Costa Rica. Many thanks to all the people joining our chat in YouTube and Facebook. I really appreciate your comments. And let's keep our eyes on the screen as we will, hopefully we will find more amazing creatures here at this summit of the Caballito Seamount. We just reached the summit, and let's be prepared because there might be some nice surprises ahead of us. Muchas gracias a las personas que veo por ahí en los chats de Facebook y YouTube, saludando y reportándose. Para mí es un honor y un placer acompañarlos en este, casi ya al final de este buceo, número 11. Y bueno, si están sintonizando en vivo desde Costa Rica o algún otro lugar de habla hispana, por favor déjenos saber de dónde se conectan, o si tienen alguna pregunta, algún comentario, pues con muchísimo gusto por acá les vamos a estar leyendo. Para mí pues siempre es una gran alegría ver si nos están sintonizando de alguna parte de habla hispana. Mientras vemos esta zona en particular que está en la cima de este monte submarino, hay muchísimas estrellas de mar. Particularmente estas son estrellas quebradizas, que se le llaman también ofiroideos o ofiuras, y estamos viendo este pez muy común de estas profundidades, mientras los pilotos nos deleitan con esta toma bastante bonita. Wow. De este, me parece que es un ofidio, un tipo de cutthroat eel, parece. Y que bueno, que en este momento se despide de nosotros. So very incredible imagery, thanks to our pilot team here of the ROV in the control room, and I would like to also give special thanks to them. So we have currently J-Rod and Mike controlling the ROV and the robotic arms, and also Alex behind the scenes making everything possible. So a special thanks to them and all the Schmidt Ocean Institute team. So we see really active brittle stars around this sediment. You can see white, small patches of white sediment, and according to our colleagues here on board specializing in the sediment and the geology, might be carbonate ooze. So this tell us more about the dynamics at this particular place, and also based on the sediment samples that we've been getting, we can tell what's the history of this seamount. Because as you can imagine, these sediments have been building and building over thousands and millions of years, sometimes even building and forming thick layers between 200 and 400 meters.
All right, you want to just keep going up this ridge?
Yeah, let's go ahead and
You want to just keep following it to this point over here?
yeah, let's try. Let's try to go like to the left and try to be not on the very top. Let's just so we can go to that position.
Can I go more to the right? Or
Yeah, well, yes, actually to the right.
I'd rather have the ship.
Okay. Okay.
All right,
Perfect.
I'll start to slide into my right, just so you know for your next move. Pick them out.
Perfect. Thanks.
Weird rock. That looked like a bag. Weird rock.
Alex, can we pull out the overall map?
Yeah,
Just
let me
to show.
toss the map up.
Yeah, I've seen it several times, but
Yeah, no, it's good.
just to for people who's wondering where we're at right now, well, we see this pagurit crab, which is carrying. Let's see. Well, it doesn't want to be seen today. Not very friendly. All right, so yeah, it's carrying an anemone. So it's definitely really fast. Maybe not that happy of our lights, but we can tell that this crab is a type of pagurit crab. Has some sort of association and relationship with the hermit crabs that we can see in the shallow waters, usually at coral reefs or intertidal rocky shores. But these ones are definitely way faster and agile. And we see what you see right now on your screen is the track and the waypoints. So WP stands for waypoint. So these are particular spatial markers that we have been following along our dive, and we started at the abyssal plane and went up towards this place that we're currently, which is the summit, as you can see. So in purple you see like more like deeper waters, and as you go through the color scale from blue to green, yellow, and red, you have in red the more shallow waters, let's say. So as you can see, this is the summit. And we're gonna keep exploring the summit, so stay tuned and let's keep our eyes wide open as we might find some surprises. And thanks to Alex, our colleague from the SOI team, who pulled this really nice map for you to have an idea of where we are right now.
Do you know when the last time was we saw rocks? Probably can't see it in the dark.
So we see on our left sea cucumber, so they're common also of these depths. Usually at these depths, what you will find the most in these specific soft bottoms with sediments are echinoderms, which are a taxonomic group composed of sea stars, brittle stars, sea cucumbers, and urchins. And another group that is not as common, but that is called crinoids. And the echinoderms are one of the most dominant taxonomic groups at these depths. So this is why you see many of them dwelling and lurking around over the sediment, sometimes even burying under the sediment. And they usually are associated with these sediments that are enriched or that are rich in organic matter that sinks from the upper water column. So if you have any questions, any comments, or would just like to tell us where you're watching us from, please put it up in the chat, and we will be more than happy to read and answer your questions. So that look like a type of cosquille, also a very common fish at these depths. Para los que estuvieron viendo el mapa que teníamos anteriormente, estábamos viendo una batimetría de el sitio donde estamos, y como vieron, estábamos siguiendo, por decirlo así, un montón de puntos identificados como WP. Eso básicamente lo que significa es waypoints, y son puntos que hemos ido siguiendo y muestreando a lo largo de este buceo. Sin embargo, una vez que hemos llegado a la cima, entonces ahorita nos vamos a dedicar exclusivamente a explorar y ver si logramos obtener evidencia de alguna de algún flujo hidrotermal, o inclusive de algún pulpo, ¿verdad? Que es parte de los objetivos principales de esta expedición. Teníamos la idea de que a esta altura, por decirlo así, o a esta profundidad, en la cima del monte, íbamos a ver rocas. Sin embargo, nos encontramos con la sorpresa de que hay mucho sedimento, así que entonces intentaremos movernos un poco más como hacia el, por decirlo así, hacia el flanco este del monte submarino, para ver si logramos ver algún sitio donde haya rocas, y esto es particularmente con el objetivo de tener más posibilidades de ver este flujo hidrotermal, si es que existe en esta zona. Y esto es ya que usualmente el sedimento lo que crea es una capa que no es permeable, y entonces que impide que el agua salga por aquí. Usualmente entonces, como vemos y como pues se tiene el conocimiento común, mientras vemos este erizo, el agua busca salir por donde encuentra menos resistencia. Entonces, pues se imaginan, no va a salir por donde tiene una capa de sedimento muy ancha. Entonces, usualmente busca salir por ciertos flancos asociados a grietas en donde está el material volcánico antiguo. Entonces, usualmente esos son los sitios que tratamos de buscar. We have reports from Hawaii and from Arizona. I hope you are enjoying this live stream as we see this beautiful jellyfish.
I would say like kits at this point would be helpful because then some of the ones where we just took a sample, I could actually open it up and take a bit of intestine. If
Okay.
it's something that we've gotten before.
Okay, yeah.
I'll leave it to you, your call. but-
Okay. All right. Yeah, it wouldn't hurt to have. All right. Okay. And we have more reports of people watching this live stream from other parts of Arizona and even from Chile. So special greetings to all of you. I hope you are enjoying this live stream. Either depending on the time where you are, the time zone. For example, here is almost 4:30 p.m. So many people in Costa Rica call this the coffee time, coffee hour. So there are different specialists and different scientists with particular interest in all the taxonomic groups. So we have people on board interested in the octopus, people interested in the octocorals, and just wanted to say that one of the main interests that myself and my colleague Holly have is particularly these sea cucumbers that you can see here. So we will be paying special attention to see if we can find any sea cucumber that looks promising and that we might not have been able to collect previously. While we watch this huge urchin, and as you see, there are many. Oh, what's that at the bottom? No, that's a relicanthus, but what was at the bottom? So that's like over there.
Those are just the white mounds.
Okay, white mounds. Okay, so we see a relicanthus, this creature that has these long tentacles. Pretty close. Probably they're friends with this brittle star. And to the right, we saw some really nice sea cucumbers. So we're gonna see if there might be of any interest for us. So, well, we have this great close-up of this relicanthus. So yeah, these are one of my favorites. This is a type of Cycropodes, sea cucumber. So they're also called several names. Some people call them sea bunnies. Some other people call them gummy squirrels. You can tell why. They are really almost like gummy textures. And these are really important animals at this abyssal depths, especially because they feed on the sediment and they have pretty impressive rates of ingestion of all this sediment. Sometimes you will see a lot of feces and traces of them being left behind in the sediment. So they move with those feet that they have there, almost like tubular feet. And sometimes it's hard to tell the difference, but what you have here, let's say below that appendage that's like a little sail, that's the posterior part of the animal, and the other part is the where they feed on. So beautiful gummy squirrels. I think that yeah, I think we might give it a try and have one of these, because we haven't got any of these. So
Okay.
yeah, we have a really.
Power box, I imagine?
Do you think we can fit it in one quiver?
Or they're gonna start trying to escape, but hey, challenge.
Yeah, they're pretty big.
Really?
Like,
Yeah.
they're long, so the quivers, I don't know if they're long enough. I mean, we could always give it a try. If it doesn't fit, then we got it somewhere else. But
Okay. Yeah, I mean, let's try if we can put it in one quiver, and if not, one bio box.
Yeah, it'd be better to save the bio box, I think.
Exactly. That's the
Something more
thing.
interesting.
Yeah.
you get that. To go in first, maybe it'll
So we're gonna try and get a sample of this Cycropodis. We have not been able to sample this before. We've seen many, but this will be our first sample of this genus. So while we sample, I will also like to send my greetings to all the people tuning in. We're getting some reports from Australia, from Southern California, even from Brazil and Argentina. So we have a very diverse audience. So special thanks to all of you for tuning in and enjoying this live stream. We have a question. If we have seen...
Yeah, that'd be good.
What was that came in that screen? Oh, jelly. Okay. We have a question. If we have seen any sea slug during this survey. So I'm not sure what you refer to as the survey. So I think in this type we have not seen any sea slug. But yesterday we saw pluribranch. I think Vitulinit. So we were able to collect that specimen, and it is going to be deposited in the Zoology Museum of Costa Rica. And this is very, very important for us because it will allow to make the most out of all the samples, because we really appreciate all the samples that we are able to collect and have back on board. They're precious to us. And also this will make possible for other colleagues around the world to have access to these materials for future investigations. And also a good time to remind yourselves and remind us that everything that's been happening as part of this Octopus Odyssey Expedition has gone through a strict permitting process with the Costa Rican authorities. So we try. Yeah, sorry. Yeah, can we put the lasers actually? Yeah, sorry, I forgot. Yeah, no problem. Yeah, okay. So we just saw some rolling action, and as I was telling you, all these samples you can call it like rolling in the deep, right? I think it was a song for this guy. I was telling you that all these samples are going to be deposited in the Zoology Museum of Universidad de Costa Rica. So it is pretty important that they will stay in their home place. So even though that, you know, everyone interested in the samples can have access, they will remain here also to be able to have that facility for national colleagues who might be interested in these groups. Right. Did we get a nice view with the lasers? Yeah.
Quite large.
Yeah, it looks large. So
It'll fit inside the equipment.
It might be too long.
Mhm. Like squish it a bit. Yeah, that's okay. I think they're pretty resistant.
Yeah. And the plan is to get the ear part in first, and hopefully it folds back.
Okay.
That way.
Okay.
Yeah, that's actually the best too for the look at sort of stuff we're going to dissect, so protect that. But
Yeah.
Alright, are we good to go in? You happy?
Yeah.
yes.
Yeah, so those green dots that you see on the screen, while we prepare to grab it with our robotic arm, those green dots help us to measure these creatures living down here. And these two green dots are separated from each other by 10 centimeters. So we can have a rough estimate of the size. In this case, this cucumber might be 30 to 40 centimeters long. But yeah, this is again just to have an idea. And when we take a closer look at all the footage and then of course at the samples, we are actually able to confirm all this preliminary assessment.
Looks like it's got a commensal worm down by the oral groove there.
Oh yeah.
Oh.
Cool. Oh no, stay there, little buddy. Stay there.
They never stick around.
Oh yeah.
Yeah. No, you'll frequently find those under sea cucumbers, it seems like. I don't know if they're just stealing little bites to eat from whatever the cuc is picking up.
So as you see this sea cucumber, we also sometimes you will hear from us that we call them cukes. It's just funnier and catchier. So this cuke is pretty large. So we are very thankful to have very skillful pilots that can actually try to see if they can fit in one of these containers that we also call these white cylinders that you see on your screen are called coral quivers. And yes, not exclusively for corals. So you see the beautiful color this sea cucumber has. Again, this is Cycropodes. And we are not sure about the species, and this is why we are collecting.
You want it to fold back on the back, or I don't know. Either work, but start working your way in there.
Así que para todas las personas que están viendo esta transmisión en vivo, estamos tomando una muestra de este ejemplar de pepino de mar. es del género Cicropotes y a ver, pues vemos que hay varios de estas muestras, así que tenemos la tranquilidad de que no estamos afectando su población. Hemos visto elevados números de estos animales, así que para nosotros es importante poder colectarlos para entender más sobre su anatomía y su identidad taxonómica.
So we do frequently get questions about collections of live animals, and it can be difficult for some folks thinking about collecting these animals. But the thing that the ROV allows us to do is be very selective with this. So although we are taking a live animal, we can be careful in our selection of it, and we can ensure that we're getting a good specimen so that we can take less specimens overall. Non-selective collections, things like dredges, those just kind of go through and take all the animals in an area and frequently will damage the animals. So you end up getting not fantastic specimens. So with the ROV, we can be a lot more selective, and although we are taking live animals, we can hopefully ensure that that animal is much more valuable and much more intact. That way, ultimately, we're taking less specimens.
Okay. Are we going BioBox 3B?
BioBox 3B?
Yeah, let's do that.
Okay.
So we're gonna go ahead and put this sea cucumber in this bio box, which is a larger container in this case. If the specimen cannot fit the other containers, then we have these other ones that allow us to have larger ones. And thanks also to Alex that just explained us the importance of this sampling, and also that we are very, very careful. Just wanted to say that I am based in Costa Rica. I'm a Costa Rican, and one of the main things here, not only for me but for other colleagues on board, is that we really, really appreciate our biodiversity. That's the most important thing for us. So believe me when I say that we try to have the minimum impact that we can have, but sometimes it's not enough with the images to get to know more about these animals. And also, hopefully in the future, based on these observations and all the science that's being done, we will have more conservation and protection measurements of these sites. Everyone, for all your comments and questions, please keep them coming, or even if you just like to let us know where you're watching us from, we will more than glad to read this here in the control room. All right. Cool. Perfect. Thank you to our pilots here, J-Rod and Mike, who did this awesome collection. So we're gonna keep exploring this summit and looking for any evidence of venting or shimmering water. As I was saying earlier, we are surprised because we were expecting to see more of a rocky substrate at this particular site. So science is pretty important, and I would say that also offers us with many surprises. And this is the nice thing about exploration, that you never know what's going to happen. What are we going to find next? Así que vamos a seguir enrumbados explorando la cima de este monte submarino llamado Caballito, y vamos a seguir buscando posibles evidencias de fluido hidrotermal o inclusive de algún pulpo que podamos ver. Al mismo tiempo también estamos con el objetivo de tomar muestras de octocorales, ya que tenemos acá a la especialista en octocorales de la Universidad de Costa Rica, doctora Odalisca Brady, que está muy interesada en entender más todas estas especies que viven en las aguas profundas de Costa Rica. Oh. ¿Quién dijo? Mientras vemos este picnogónido, is feeding on something. Esto se le llama también araña marina. Pues claramente no son arácnidos, pero pues se les llama así por esa forma que tienen. Ah, tiene otro. It has another one. Oh, wow.
Oh yeah, it is holding something there.
Another one. It's another picnogoniid. Okay.
Yeah. But it's carrying.
This could be mating or something.
Oh. Can you annotate that? I don't know.
No, it's got its proboscis out.
Okay.
So in the middle there, and it looks like it might have one of those radiolarian things that we saw rolling
Radiolarian.
along the bottom the other day.
Mm-hmm.
At least that's my reading of it.
Okay. Wait, this is the radiolarian. Maybe we can collect it all the same.
I mean, this guy's huge.
We've got a big bio box. Take the food out of his. Yeah. What's the typical look for it?
Sí, parecer se está alimentando de algún organismo como
I don't know if
gelatinoso.
it's one of those radiolarians or
No,
not.
I think it's like a tunicate, no?
Or a ctenophore.
Maybe a ctenophore? Yeah.
Yeah, I mean,
Entonces esto que vemos aquí es un picnogónido, que también se le llama araña marina. Viven usualmente o son bien grandes a estas profundidades, e inclusive se han encontrado en aguas polares. Y bueno, a pesar... No. Yeah, no, let's keep looking. Yeah. No, ya estamos en lo más alto. Lo que vamos a hacer ahora es hacer esto. Sí, como ver si podemos irnos acá. Porque parece que todo eso rojo va a estar como sedimentado. Entonces andamos buscando a ver si encontramos como más rocas. And as I was saying, that we just saw was a picnogoniid, also called sea spider, and it is definitely not a spider, but very similar look. And we have a question in our Facebook chat about the vibrant colors at this depth. So the question is that if there is no light, what is the evolutionary purpose for being so colorful? So that's a great question. Pretty much what we know so far is that most of these animals have this colors, usually different tones of red, and basically because red is the first color that's being filtered through the water column in the oceans, So that means that animals being red at this particular depths are almost works as a camouflage for them. Oh, what's that?
Dead or a molt? It's not intact, so it makes me think maybe not a molt. A lot of times molts will be pretty good, but I don't know.
Well, we're seeing, yeah, some sort of that squat lobster. We have a colleague suggesting that is a molt. Who knows? And yeah, so I was telling you the evolutionary purpose of this colorful tones that we see on many animals is actually a camouflage, because if they're red, since red is not, it's being filtered in the first hundreds of dozens of meters of the water column, that means that at these depths they are almost invisible. Let's remind ourselves also that at this abyssal depths there is almost total darkness, except for the light that's being produced by the bioluminescent animals, but apart from that you have pitch darkness. So these colors help them to, again, camouflage. And even some of them, and we've seen this let's say features and some sea cucumbers that have these bright colors, that they are also sometimes show bioluminescent patterns in their behaviors.
Yeah.
So thanks for the question, and if you have any comments, any doubts of what we're seeing, please let us know either through our Facebook or YouTube chat.
Yeah, we saw one earlier that we thought was dead, and it was intact, and my head went, maybe it was a molt. This one, like, the legs were off, and I'm thinking of all the crab molts and shrimp molt and that sort of thing I've seen, and yeah, the legs are usually on there, so that one could have just been a dead thing that got predated on, but no real way to know other than to pick it up and get it up here and be like, is there meat in there?
Have you ever seen a live one yet?
A live squatty? Not on that one squat there.
That was
Like a little slipper lobster or something. Yeah.
It looked like someone's lunch.
But we haven't seen a live one yet, right?
We actually did see some large squatties and other crustaceans on the way up. I don't think since we've gotten up here we've seen any walking around. I am actually going to be resetting the streams, restarting the streams here shortly. So this stream will end on both Facebook and YouTube, but a new one is coming. We're not done down here yet. So just keep an eye out. It'll take a couple minutes to get the new ones up and going, but we're going to restart here shortly.
Nada más para comentarles lo que estaba diciendo mi colega Alex del Schmidt Ocean Institute acá en el cuarto de control, es que debido a cuestiones de espacios en las diferentes plataformas se va a reiniciar la transmisión, así que no piensen que estamos terminando, sino es nada más para poder alojar estos videos en Facebook y en YouTube. Entonces seguramente se va a suspender por un par de minutos, pero rápidamente vamos a estar de regreso, así que quédense atentos porque por acá vamos a seguir explorando.